position of Vice President, Pharmacy Council of India as our guest of honor. We are also privileged to have respected Dr. V.K. Maurya, sir, Principal, Government College of Pharmacy, Aurangabad, as the president of today's function. I now request respected chief guest, Colonel Venkat sir, to declare the opening of STTP program. Sir, just a sentence we require from you that the program is open now. I request Navle sir to please raise the curtain. Thanks a lot, sir. I now request coordinator of this program, Dr. R.B. Navle sir, Associate Professor, Government College of Pharmacy, Aurangabad, to brief us about the program.
Thank you, madam. Am I audible, madam?
Yes, sir.
Thank you very much, Mayura, madam. Good morning to all dear friends and participants and my colleagues. I, Dr. Rajesh Bhaskar Navle, coordinator of this AICT sponsored 1 week short-term training program, wholeheartedly welcome you all. I want to extend my sincere thanks first of all to the AICT for providing us this opportunity and grant for the conduct of this beautiful program. First of all, on the behalf of Government College of Pharmacy, I welcome Colonel Venkat sir, Director of AICT, and Dr. Shailendra Saraf sir, Vice President of PCI New Delhi. Thank you very much, sir, for accepting our invitation and to be as a chief guest and guest of honor and present for this inaugural function. Dear friends, I congratulate all you to that you all are going to be witness and part of this event, which all of us will enjoy for a week. Little bit, I would like to brief you regarding this 1 week STTP. The theme of our program is Advancement in the Pharmaceutical Research, a journey from the academics to the industry. Dear friends, this 1 week shorter program will primarily focus on introduction of principles of smart research. It will also focused on the formulations of the problem and then the solution of these problems to maintain the perpetual integrity of the data. Dear friends, I'm confident that after completion of this 1 week STTP, you will all will be acquiring the knowledge, skill, and attitude in the domain of research, organization of study in clearly defined components or phases, training and upskilling of the faculty in use of the various relevant advanced research methodology, and improved industrial collaboration for the research mobilization. In short, these are the expected outcomes of our short-term training program. Dear friends, we along with me and our team members will be with you all during this program for the 6 days, from today morning till the 29th of evening, that is up to the Saturday. I'll brief you regarding how this program will conduct during this 1 week duration. We have already created a WhatsApp groups that is AICT STTP B and AICT STTP B1. All of you have already joined this group. Please join if you are not. Requesting all to that you will be in one group only and not necessary to be in the both groups. This WhatsApp groups will be official way of communication between you and us. I have already put the detailed schedule of this program on this WhatsApp group. The program schedule will give you the idea how this program will run during these 6 days. Our pre-lunch session will begin at 10 a.m. and the post-lunch session will begin at the 2 p.m. sharp. We will start our program morning at 10 a.m. with the prayers. So we are requesting you to join Zoom well in advance at around at least by 9:50 a.m. The link for joining this will be provided to you on the daily basis on WhatsApp group at least 15 minutes before the commencement of session. Our lunch break will be of 1 to 2 p.m. Afternoon session will start at 2 p.m. sharp. To avoid any delay in the afternoon session, I request all of you to join before 2 p.m. We will open these links sharp at 1:45 p.m. for your login. At end of the day, you all will have a assessment of for the day. The Google link will be provided to you on your WhatsApp group. All you have to attempt this test immediately. The test will have 10 to 20 questions, and outcome of this test will be counted for your overall evaluation purpose. Dear friends, as an AICT policy for the certificate, you have to attend at least 80% of the session, and you have to spend at least 80% time in that session, and you will have to score minimum 60% of the mark in online test. Your attendance will be taken care by the Zoom app. Our app will monitor your incoming as well as outgoing timing automatically. I'll request you to log in with the Zoom with the same name and email IDs, which you have already given to us during the registration. Log in with the same name and ID in all the days. Dear friends, it is creating a lot of troublesome or consume a lot of time with the multiple ways of name entry and email IDs to consolidate this data. So it will create an unnecessary delay in the issue of your certificates. So I request all of you to be in the group with the same name and email ID, which we have given us during the registration. To the all participants, I request you kindly maintain the decorum of the meeting by putting your mics on the mute basis and on it whenever you required it. And also on the WhatsApp group, we will operate the no good morning policy, that is no wishing message or no smileys, etc., so that the message will be eventually fully communicated to you properly. Dear friends, me and along with our team is always with you to resolve any kind of your queries, difficulties, and doubt. You can directly contact me or any of our persons in the team at any time in person. You are once again welcome to all for this 1 week STTP. Wish you a happy learning. Thank you. Thank you very much. Over to you, Mayura madam.
Thank you, sir, for briefing us and the participants about the objectives. I'm sure that all the participants will be benefited from the outcomes of the program. I now request Dr. S.B. Bothra, sir, Professor, Government College of Pharmacy, Aurangabad, to introduce us to today's chief guest, respected Colonel Venkat, sir.
Good morning, everyone. Am I audible?
Yes, sir.
Good morning. Good morning, Colonel Venkat, sir, Dr. Saraf, sir, Dr. Maurya, sir, and all our team members and all the participants. Good morning, everyone, and welcome to this gracious function. It is a great honor and pleasure to introduce such a multifaceted talent and disciplinary person, Colonel D. Venkat, who is a serving Army officer from a technical branch, that is Corps of Electronic and Mechanical Engineering. We have done his mechanical engineering from Army institutes, and thereafter he has served nearly 30 years of his career in various positions in commands of staff and instructional across the country and abroad in the military. The officer is presently working at AICT, and he is handling faculty development related issues, particularly related of grants through various AQAS schemes, that is quality improvement schemes. The officer has a unique distinction of representing India at its technical military attached to Russia. The officer also handles important quality initiatives like NITTT, that is National Initiative for Technical Teachers Training, which is mandatory for the purpose of regularization and promotion of all teachers. Particularly, I must say I'm very proud to say that few people from our college, we have attended the mentors training. It was the first mentor training where myself, principal sir, and Dr. Navle sir, we all have attended, and it was very excellent training, which was done under auspices of NITTR Bhopal. And Dr. Venkat sir was very, very instrumental and very keenly observing the developments which were going on there. The implementation of examination reform also on his cards. AICT student learning assignments, 360-degree feedback, and process for implementation of National Education Policy 20T0. These are just few glimpses of what he is doing. But beyond that, many might have witnessed that he is a man with discipline, man with technical details, and therefore he is very keenly trying his best to improve the quality of technical education through various approaches. With this brief introduction, may I request respected Colonel Venkat sir to kindly guide us, kindly bless this occasion with his good words.
Dr. Sunil Batara sir, thanks a lot. And it's absolutely pleasure and absolutely honor that, I'm on your platform for the purposes of inauguration. And it's a great honor that you bestow upon me that I find myself here amongst one of the best on the particular subject. So your college does not require any large introduction. The introduction itself is so very evident when I told that I'm amongst one of the best, and I do feel myself very elated to be here. So thank you for that. Thanks a lot to the Government College of Pharmacy, Aurangabad. It's an absolutely pleasure to be here and to take it on that you people are carrying on an AICT sponsored STTP program of advances in pharmaceutical research, and that too from academy to industry. You will be showcasing the path from academy to industry, and that is what is the need of the day, and that has not to be spoken again and again, as you have proved yourself in the last year and the year in which we are living now. Not only that, now it's from the history that you people have been proving, and the coming year and the coming times in which all of us are staying has just strengthened our beliefs in various pharmacy management, the staff, the students, the institutes. So you have actually speaking cemented the belief that behind everything what has happened and the issues which each one of us was going through, the solution lies within, and that is you people. If we have one of the best minds and we have one of the best solutions, and if AICT can continue to remain associated, I don't think so that there can be a better opportunity for AICT, because today pharmacy colleges are one of the esteemed colleges of India. The institutions are so very adored, that they are so very respected, that everybody likes to get associated. And quite obviously, in spite of issues moving around, if your institute continues to be associated with AICT, let me tell you, it's not your gain, it is totally AICT's gain. That we get to know one of the best people, and the people who are associated today as part of various participants and as part of various attendants, and what an opportunity. I think you're not going to get an opportunity better than this ever. These are the times, these are the opportunity, these are the places wherein, in case if you get connected to the best minds on the subject, I think your career path is already well charted out. So congratulations for an absolutely wonderful work. Congratulations for coming forward and continuously being on the subject of ensuring that you are imparting knowledge to your fellow colleagues, and you are imparting knowledge to various other faculty members and research scholars who are being associated with you. Every time you do this program, there is always one subject which comes into mind, and the subject which comes into mind is, the more you actually speaking organize events of this nature, the more you come on platforms and speak, the more connected you feel, and the more knowledgeable that sharing of the knowledge takes place between you and the various participants. So to that way, I congratulate the coordinator, I congratulate each and every staff, I congratulate the management, and to top it all, the principal, sir. I'm very much honored that actually speaking, a program which is supposed to be conducted. We have the principal of the institute also connected. We have the principal driving this particular program right from his forum. And when a leadership gets involved in any activity, I don't think so we need to say anything more. Then the results are very much there to be seen. And that is what I was trying to tell in today's days when the participants are logged on to your platform. There are many things that they're learning. These are one of the side aspects of an online teaching and learning, wherein because of your conduct in an online platform, your reach now has gone way, way beyond Maharashtra. You are not limited to within one geographical region. You are actually cutting across the complete length and breadth of the country. Everybody is focused on you, not only on your academic brilliance as to what is being spoken. People are also focused on the conduct mannerism in which it is being delivered and it is being presented, and the effective utilization of the technology. Everybody wants to learn, everybody wants to improve, and you give a huge chance to a lot of us to learn better ways and to learn how to present themselves. Congratulations, thank you, well done. Do let us know if AICT can be of any assistance at any given point of time. It would be our pleasure and honor. Thank you.
Thanks a lot, sir. Thanks a lot for your, first of all, appreciation and kind words and your motivational talk. We are definitely motivated and we will meet up to your expectations surely. Thank you, sir.
Thank you, Mayura ma'am. Thanks a lot.
Thank you, sir. I now request Dr. S.R. Shahi, Madam, Associate Professor, Government College of Pharmacy, Aurangabad, to introduce to us today's guest of honor, Dr. Saraf, sir. Over to you, Shahi, Madam.
Thank you very much, Mayura Madam. I am honored also, and it's my pleasure to introduce my super senior, Professor Shailen Saraf, sir. अगर सर के बारे में बोलेंगे, स्टार्टिंग वर्ड तो है, ही इज alumni of Department of Pharmaceutical Sciences, Sagar, and a student of a great teacher like Professor V.K. Maurya, Dikshit sir, N.K. Jain, many to add to the list of Sagar teachers. Professor Shailen Saraf is a pharmacy professional who is actively involved in promoting pharmacy education and research. He completed his graduation, post-graduation, and doctorate in pharmacy from Dr. Hari Singh Gaur Vishwavidyalaya, Sagar, in the year 1986. That's his graduation. 1988 is PG, and 1993 is PhD. He, talking about his professional journey, he served as a professor and principal at B.R. Nahata College of Pharmacy, Mandsaur, MP, in the period of 1995-2002. Then as a director at the University Institute of Pharmacy and Dean Faculty Technology at Pandit Ravi Shankar Shukla University, Raipur, CG, that's PRSU, for more than a decade. He also served as a chairman, Board of Studies in Pharmacy, along with most of the prestigious statutory positions in PRSU. He was also a Vice Chancellor of the State University, Hemchand Yadav University, Durg, Chhattisgarh. He has been working very closely with the PCI for more than 16 years and currently holds the position of Vice President of PCI. During his professional career, he was associated with various national, statutory, regulatory, and accredited bodies like PCI, AICT, UGC, DST, NAC, NBA, and many more to add to the list. He has significantly contributed to the pharmacy profession through different professional bodies like APTI, IPA, ISP, IACP, AASP, ISCA, ISTE, and he was also a member. He's also a member of International Commission of ACPE USA. As a researcher, he has worked in the field of herbal technology, medicinal plant research, and NHDDC for the last 3 decades and successfully completed several research projects supported by extramural research funding agencies like UGC, AICT, MPCOS, CGCOS, DST. He continues to play a vital role in establishing and implementing several institutional projects like UGC SAP, AICT MODROP, DST FIST, and DST supported National Center for Natural Resources. During his tenure as a director, that's a period of 2002 and 2013 and 2016 to 2017, the University Institute of Pharmacy of PRSU, thus Raipur, was recognized one of the best utility departments, NIRF ranking 48 in the year 2018 of the PRSU, and a nationally recognized center for research in pharmaceutical sciences. Today, he supervised 15 candidates for PhD degree and contribute to more than 200 national and international publications in different peer-reviewed journals of national and international repute, authored for 7 books, chapters, filed 10 patents. He has a citation of 4,100 citations and 30 HI index in Scopus. He's also been a reviewer and the member of various editorial boards of different national and international journals, and served as a chairperson, resource person, and keynote speaker in several national and international seminar, conferences, and workshops. His exemplary work has been recognized with several awards: Fellow of International Institute of Organizational Psychological Medicine, USA, Australia, UK in the year 2019. Dr. K.R. Parikh Award, ISTM India 2019. Platinum Jubilee Lecture Award, Indian Science Congress, 105 ISC Imphal, 2018. Merit of Excellence Award, ISP 2018. Lifetime Achievement Award, Pharma Ratna Award, RDM New Delhi, 2018. Late Smith Kamla Ben Vavia Memorial Award for the contribution to APTI, 2017. Pharma Ratna Academics Administration Award, 2017, New Delhi. IPA Fellowship Award, Indian Pharmaceutical Society, 2016. Principal of the Year Award, APTI, 2016. Diamond Jubilee Illustration Alumni Award, Dr. Hari Singh Gaur, University, 2016. Golden Jubilee Illustrious Alumni Award, that is again Dr. Hari Singh Gaur, University, 2006. PD Sethi Best Paper Award, PD Sethi Annual Award 2010, Best Paper Award APTI in 2008, and Best Paper Award in IPA, that's IPC Hyderabad 2005. These are just a few. I have made a mention of the various field, but sir is very actively and dynamically contributing the field of pharmacy, and always on his toes. Whenever we call him and we want any support to that level, he always supports. He has traveled different countries in various capacity. That to name some is USA, UK, Switzerland, Singapore, Malaysia, UAE, and Mauritius, etc. And I'm proud to tell he is my senior when I was in first year. और एक पर्सनल टच ये बोलना चाहूंगी कि जब हम फर्स्ट ईयर में मतलब फर्स्ट ईयर की बात है, वी आर ऑलवेज केयर्ड इन द डिपार्टमेंट. ही वाज ऑलवेज अ मेंटर. हमें कुछ भी प्रॉब्लम होता था तो हम सर के पास जाते थे, सर बता दीजिए, सर बता दिए. सर वाज ऑलवेज मॉर्निंग, अर्ली मॉर्निंग, आई डोंट नो व्हेन ही वाज कमिंग बिकॉज वी वर गोइंग आफ्टर 10, and late evening also, even when we were leaving the department, he was still there. So I don't know what time he was coming and what time he was going, but 24 बार like कभी भी सर को कुछ पूछा, कभी भी डर लगा तो सर को जाके बोल दिया, सर ये नहीं समझ में आया, ये नहीं समझ में आया. आई वुड लाइक टू गिव हिम अ टाइटल एज सीनियर फार्माकोपिया. तो हर चीजों के लिए उन्होंने हमारे समस्या का हल भी किया है, एंड ऑलवेज कॉन्फिडेंट एंड मेंटरिंग. So thank you, sir. On behalf of the college, I welcome you to this platform. Yes, Mayura madam.
Blessed to have such an iconic personality as guest of honor. May I request Dr. Saraf sir to share his valuable words of advice. Please, sir.
So I think my screen is visible.
Yes, sir.
Okay. So thank you very much, Sadhna. You really make me nostalgic this morning. It's really my proud privilege and honor to be here on this platform this morning. And the only two lines were sufficient, first two lines when you are giving my introduction. I'm alumni of Saga University, and I have this honor to be there and interact with the teachers like Dr. Maurya and Dr. Dikshit. I'm really brought you the greetings for this morning, and Honorable the principal of the institute, my own teacher, Dr. V.K. Maurya sir, the guest from AICT, Colonel Venkat, Dr. Sadhana, Dr. Mayur Kale, and other teacher friends, and most important, the participant for this STTP. It's a I can say, though you have made me nostalgic, I'm just feeling like thrown into memory lane, but still I have to honor my responsibility. So I'm just giving overview about the topic and necessity of such type of activities, which are very much needed during this time. So we have seen the toughest time of our life, but after this problem is coming over gradually and slowly. So what we are thinking about, no doubt we have suffered a lot, but those who are surviving are more wiser than they were in the early 2020. So I have just decided divided this particular time on the basis of digital divide. And digital divide is a concept where two sides of the things are entirely different. And we are fortunate to witness this particular event. As far as I'll begin with the light of it, and that is during the pandemic, only thing which was beating the rising graph of corona was webinars. The word was not known before this pandemic, but we have seen how we have transformed and we have started organizing webinars. But we have also observed that when we have developed the competence of sleeping in the lecture halls while your eyes are open. Now this particular webinar, this after corona, this period is providing an opportunity. While you are sleeping, you can attend the webinars. So this is just only as a light point, but we have to understand the things what are happening all around. And for this, there is a need to understand what is really happening in the profession. So there are a lot of things we are told before this corona. India is a pharmacy of the world. India is the supplying drugs to the over 160 countries, and we had several things. But we have also able to identify the paradox situation in this during this particular pandemic, when we are patting our back for this supplying of the drugs for more than 150 countries, 60 countries, but the real heroes were 3-layer mask and sanitizers. We were not able to provide any facility as far as the infrastructure is concerned and medical health infrastructure concerned. We were told through McKinsey and other survey that India is the preferred destination for medical tourism. But what was the reality when this pandemic has came? So we were not able to provide the sufficient beds also. And even when it is mentioned then that there is sufficient high-end medical facilities are available. So we have again come to know that these facilities are just available at the very posh locations in the country. And during this particular pandemic only, we have come to know, we have realized the existence of different concepts like artificial intelligence and how this role is going to be of a pharmacy going to be changed in times to come. What the times task ahead? So this is during this particular time only we have observed there is a worldwide, we can say, paradigm shift from treatment to prevention. They are coming back to nature. There are a lot of 3D printings and all that. And even in the field of research also, the total landscape of research is changed. People are not preferring to go to wet lab. What they are saying that a lot of things are available. We have to properly have done the data mining. We have to do the data analysis, and with the help of computational facility, there are research like microbiome activities, laboratory and clinical trials. It is worthwhile to mention here only USA, where only 5% population of the world is more than 27% clinical trials are carried out. But in India, we have only 2 to 3% clinical trials, and we are having different regulatory clutches. So we are just missing the new drugs inventions. But during this particular pandemic only, we can proudly say that we have transformed into a research-oriented country, and we have produced our own vaccine as well. And as far as the trends is concerned, so I'm just continuing with the all such things. Cognitive computing is there, and friends, when this particular time is ruled by the artificial intelligence and data mining, the way we will, the moment we are going to acquire the minimum workable competence in the artificial intelligence, there must be another science will be the other facilities. This is known as quantum computing. So we have to keep on learning, and this is the only thing if we want to survive. As far as the landscape of research in industry is concerned, so they are also totally transformed. And as I can mention here a few examples for the easy understanding, the Pfizer has created, instead of a big infrastructure, they are coming out with the centers for therapeutic innovations. And they are somehow trying to correlate, coordinate with the academics, and they want to utilize the best of both the things. They are extraordinary, best of state of all the facilities, but they are looking towards the academia for the new ideation. As we have observed in the West, whenever anybody is coming out with new drug, new vaccines, they are proudly mentioning that they are associating with one of the university. So this is also a very important concept we have to underline, we have to flag this. There must be some time when industry will say that I am associated with institution, and I am sure under the leadership of Dr. Moria, this particular thing will be started from your institution, that industry will say we are coming in a big way because we have collaboration with industry institution like GSP Aurangabad. Similarly, I'm mentioning even GSK also, they have also converted into small units. So the specific tasks are given, and they are trying to better utilize the specialized knowledge and less infrastructure and then investment in the research. As far as the new approach of the partnership is concerned, so this is a beautiful example. One can Google from the internet, and that is Mandi Pharma. This is the we can say a coordination of more than 100 companies which are operating in 120 companies, and they are coming out with a new drug, antifungal drug, and this is based on the sharing of the informations. And at the same time, in order to cut the prices, high investment in the research R&D. So what they are saying, you have to learn to share the knowledges, and we have to ensure that whatsoever contributing will get the due. So if we are somehow thinking about the linking academics with the industry, which is the topic of this particular STTP, we have to understand the trend of the world. We have to understand the practices followed by the industry and gray areas which are to be mapped and which are to be underlined. At the same time, points to ponder. What is required? There is always a need to have understand the gap between industry and academia. Pharmacy is a highly technology-driven profession, and the half-life of this technology is always very short. So what is required? Because there is one of the social scientists has mentioned, the teachers are in the, we can say, toughest time for the teachers, because they have to prepare the graduates in such a way that they are able to solve the problems of the future of the society, and that too with the help of tools and techniques not yet invented. What it means? Because the half-life of the change in the technology, in the practice, in the tools and techniques is so fast, and the problems are not known. So we have to prepare graduates in such a way that they immediately realign themselves and come out with the solutions in the society. So there is need to understand the practices in the industry. And as far as the syllabus is concerned, curriculum is concerned, this is the practice since the beginning. Always syllabus are designed considering the average competence, average learning capacity of the graduates. But over and above the syllabus is never ever a bar. So this is the responsibility of the teacher to identify the slow learners, fast learners, identify the practices and gaps in the industry, and accordingly give the skills to the students. And as far as the computational techniques is concerned, so we were not teaching in our institution effectively. But this is time we have to understand how to go for data mining, how to use the artificial intelligence and machine learning if you really want to come out with the solutions, and what is required from the teachers. So we are living in a different era, as I have mentioned earlier also, And this particular time, we are living in an era where we are realizing the industry 4.0. And unlike previous three industry revolution, this revolution has come into the being very silently. But the beauty of this revolution is this is amalgamation of physical sciences, biological sciences, and IT. And if you want to survive, if you want to produce something, this is your responsibility to learn the basics of the technologies and at the speed, fast changing speed as well. Otherwise, you will be left behind. So this is time when we are talking about the 4.0 revolution in the industry. This is time to adopt the teaching learning paradigm that is 4.0, and this is known as learning, unlearning, and relearning. What you've learned earlier, you have to learn this art to forget each and everything, format your learning, and relearn what is required, which is contemporary, which is the need of the industry. And at the same time, there's need to have the concept of lifelong learner, because if you want to produce lifelong learners from your institution, this is the first responsibility of the teachers to become lifelong learners, because India is also going to face very reality of the national education policy, and again the change agent of the change will be the teachers. So you have to take the responsibility, you have to take the responsibility, you have to realign yourself, and you have to start working in this direction. So again, when we are talking about the research from academia to industry, so we have to very carefully understand the concept of honest behavior in academia. We have to take care of plagiarism, fabrication, falsification, and data integrity and quality publication, because distracted publications from Scopus or magazine from this country. There is always problem of the data integrity. So whatever you are publishing, whatever you are observing or in the laboratory, you should learn to produce without any modification, and your result should be reproducible always, because somehow the publication or research is directly reflected with the connected with the respect of the country. So as far as the profession is concerned, we have to think about all such things, and we have to adopt one more mechanism that is known as seamless transfer between industry and academia. And the industry, recently Pharmaceutical Council of India has launched a scheme where they are funding the academia to go and work in the industry. And after that, we are sincerely churning on certain issues like how we can attract, we can depute the industry person in the academic institution as well. And the need, as a Pharmaceutical Council of India, we always talk about the minimum infrastructure. But again, this after Corona, in the digital device, the things are changed. So we are talking very high about what is the internet bandwidth available with, and what is the speed. And learning management system is having with the is there in the institution not. And the most important thing is content generation. So if we are teaching, we are asking our teacher to learn the basis of content generation, then they will become the true leaders in this particular world. At the same time, institutions are supposed to be ready with the storage, cloud sports, and all this data security reasons. At the same time, they should have an effective internet also in order to provide the maximum facility to the students. So I can conclude my observations about this particular seminar, this webinar, and through STTP, because this is a contemporary topic. I was just thinking to say a few words, but after visiting the website and seeing the theme of this institution, it made me to churn and come out with our specific ideas about this particular topic. And one more important thing, before this particular digital device, before pandemic and after pandemic, we have realized we were working in isolation. And each and every pharmacist is doing. So this is need, and this is the concept of the Shushrut Ekam Shastra Madhyano Namadyata Shastra Nischayam Tasmat Bahushrut Shastra Bijaniya Chachikitsaha. A physician who learned one science only cannot be sure of his own science. So this is time to learn the multidisciplinary research, and really this is time when nobody is talking about multidisciplinary research, but they are talking about interdisciplinary research. Who is able to contribute can be on board. And the same theme is also underlined by the UN Sustainable Group skills among the graduates. So we are living in a different era. We are in a second time zone of this particular digital divide, and we have realized we are connecting with the through the internet, but very soon teacher must be in place, because there is no substitution of one-to-one classroom teaching. There must be blended learning 20-30%, but nobody can replace the teacher. Nobody can replace the existence of the teachers. So while we are effectively working on webinars, first we have to remember someday we have to go to lab again and we have to work again. And this is my message for all of you. You should be adaptable for all the changes if you want to survive. With survival of fittest is not at all workable in these times. Those who are adaptable for the change will survive. So with this message, I will conclude my presentation, and I think I will expecting I'll take only 10 minutes. So I have completed my presentation during this particular time period, and I will share my presentation also if somebody wants to. Again, I will thank my teacher, my colleague, my junior, Dr. Sadhana, for inviting me and giving the opportunity. And I think this particular session, STTP, will highlight the importance of ideation, because everything is there, but ideation is missing. Albert Einstein once said, the ideation is the highest form of the research, but ideation is missing. Why I am saying so? Not only in India, anywhere in the world, no pharmacist has predicted pandemic like that. But there are some thought leaders who have predicted a pandemic like this. Even Bill Gates in 2015 in a TED Talks has compared a cloud of a nuclear weapon with the influenza virus. So they were predicting. So this is time when you are working in the laboratory. At the same time, you have to work in the ideation, and you have to start respecting your own ideas, and you have to start working right now to translate your ideas into reality. Because in our lifetime, we have enjoyed the status of pharmacy of the world, but in our lifetime, we want to see India as a research laboratory of the world, and this must be the most satisfying thing for all of us. And efforts of each and every will be acknowledged, and at least we must have a satisfaction that we have contributed our sincere efforts, our small efforts, to make this beautiful planet more beautiful for generations to come. I am really thankful for giving this opportunity, and I can understand a tough teacher like Dr. Moria is here. He used to give, it's very difficult to get the passing marks from him, but I believe today he will give me passing marks. I'll be obliged, and I'll be remain connected with all of you. It's really my pleasure. Thank you very much. Wishing you all the very best. Stay safe and stay active and productive as well. Thank you very much.
Thank you, sir. Thank you for guiding us about the planning and critical thinking and the need for the future research from the perspective of pre and post pandemic era. Also, you have given the overall expectations which are required from the teachers in the coming future. So with this, may I request Dr. V.K. Moria, sir, the president of today's function, to express his views on the theme of STTP. Please, sir.
Respectful wishes to the Honorable Dais. This program has become very much vibrant and left very less space for me to speak, because of the reason that two such stalwarts are present on the dais that before them to speak anything beyond it will not be justifiable. I remember my days when one of my classmates, probably Dr. Saraf might be knowing that Pratibha Mishra, his brother, General Rajiv Mishra, when I met him at Devdali, then we were asking something with him that how your army life is there. And that time we have crystallized one idea that what is the biggest motivation one can give in our country, then he said that the biggest motivation, if you have to see the example, then the example is the army, where the commanding officer, he motivates the soldiers in such a way that they are ready to sacrifice their life for the country without thinking that what will happen to the family. These feelings still are there in my mind, and probably in my lifetime, I owe greatest respect to the army personals. And that has motivated me to invite Colonel Venkat for this program, not only because of the reason that he belongs to AICT. My virtual meeting couple of times with him regarding RFID or the projects, the way he spoke has impressed us like anything. And probably he is the one who is having the toughest challenge to streamline the internal working of AICT regarding the project funding. Very few of us are knowing that he is the key instrumental person who is working for National Education Policy 2020. Sooner we will come to know more of his capabilities, the things to which he has outlined. It was in a sense of an engineer, army officer, and the AICT personnel, and he has deliberated his expectations that how the things should be there in the area of the pharmaceutical research. And he has also explained about the commitment of AICT towards the technical education. Thank you very much, Colonel Venkat, for your inspiring words. Beyond doubt, the biggest motivation for me is that I am the alumni of one of the prestigious institution in pharmacy in India and on global map, that is the Dr. H.S. Gore University, Department of Pharmacy, from where many leading stalwarts have made their career in education and industry. Today, one of the example is in the form of Dr. Shailendra Saraf. It is his modesty that he respects us so much, but Dr. Saraf, I am very sure that the horizontal and vertical thinking you have, you deserve these positions. You have got a very big responsibility to shoulder to shape up pharma education in contest to the global changes in thinking and requirement. You have outlined them very well. Thank you very much to accept our invitation in such a smaller time frame. This program is coordinated by Dr. Rajesh Navle and my team. They are working very hard to provide you the academic feast to think new in a smarter way to shape up your research. We have many constraints in research in academic institutes, and hence our outputs are limited. Our expectations are different from the system, and the society's expectations from us are different. Resource crunching is always there. Apex body like AICT and PCI, they are trying to help us. As rightly mentioned by Dr. Saraf, that Corona has provided us a chance to rethink and reshape our way of learning and teaching. Great cartoons here shown, they have many messages. Sleeping during webinar is one of the funny part, but I looked at it in a slightly different way, that you can learn even when you are on the bed. So this is providing us a great opportunity to hone our skills and hope that all parties will take the content in which they are interested from this particular STTP. I pray that every one of us should be benefited from this program, and we should be there to learn more things which are useful for our teaching learning from such programs. I wish every success for this program, and I want to convey my very personal regard to both the guests, team, and participant for this event. Namaskar.
Thank you, sir. Your advice is always welcome, and we continuously like to hear from you again and again. I now request Ms. Sheetal Chauhan, Madam, Assistant Professor, Government College of Pharmacy, Aurangabad, to express the vote of thanks.
Thank you, Madam. A very good morning to all. Am I audible, Madam?
Yes, madam, you are.
Yes. Yes.
Okay. So appreciation, gratitude is a very wonderful thing. So on behalf of Government College of Pharmacy, Aurangabad, it's my privilege and honor to deliver vote of thanks in the inaugural function of AICTE-sponsored one-week short-term training program on advances in pharmaceutical research, a journey from academic to industry. At first, I would like to extend a very hearty vote of thanks to AICTE on behalf of my team GCPA for providing funds to execute one-week training program. We would like to express our sincere thanks to Dr. Abhay Vaskar, Director, BT Mumbai, and Shri M.D. Shivankar Sir, Joint Director, Aurangabad, for granting us permission to organize the event and being with us all the time as a strong support system. I would like to extend a special thanks to Honorable Venkat Sir, Director, Faculty Development Cell, AICTE New Delhi, and respected Dr. Saraf Sir, Vice President, Pharmacy Council of India, for accepting our invitation of inaugural program and blessing us. I would also like to extend our sincere gratitude to our beloved Principal and President of the program, Dr. V.K. Moria Sir, for guiding us time to time. I would like to thank our program coordinator, Dr. R.B. Nole Sir, for doing the Herculean task and coordinating the overall event successfully. I would also like to thank our steering committee, Dr. Mahapara Sir, Dr. Kaya Madam, Dr. Devkate Sir, and other program coordinators, Dr. Bosa Sir, Dr. Shamkur Sir, Dr. Shahi Madam, Dr. Pathan Sir, and Jadhav Madam for doing the task punctually and efficiently. I also take this opportunity to specially thank to all the principals of the different colleges for granting permission to their faculty members to join the online one-week short-term training program. And last but not least, I'm very happy to extend my sincere thanks to all the participants and speakers for their enthusiastic participation and for being available by managing their schedules for making the training program a successful event. Once again, thank you one and all. Thank you. Thank you. Over to you, Madam.
Thank you, Sheetal Madam. I now declare that the inauguration function is over, and we will immediately start with our first session of STTP. Our honorable speaker, Dr. Praveen Patil Sir, is already there online. So it gives me immense pleasure to introduce him to you. Dr. Praveen Patil Sir is working as Head of Department and is Associate Professor, Department of Pharmaceutical Chemistry at H.R. Patel Institute of Pharmaceutical Education and Research, Shirpur. He has completed his PhD in 2014 NMU Jalgaon, completed his M.Pharmacy in Pharmaceutical Chemistry from NDMVP College of Pharmacy, Nasik, and B.Pharmacy from R.C. Patel College of Pharmacy, Shirpur. Sir has received research grants, total 76 lakhs as principal investigator from various agencies, funding agencies like AICTE, SERB, that is DST, ECR, and ICMR. He has to his credit about 80 publications in national as well as international journals of repute, with cumulative impact factor of about 130, that is from Thomson Reuters. He has filed 4 patents, to his credit 6 international book chapters in Taylor and Francis, Springer, as well as elsewhere. He has received the National Level Best MPharm Thesis Award in 2014-15 for thesis entitled Green Synthesis of Graphene-Based Biosensor for Diagnostic Applications by Rajni Bhai B. Patel Trust under the patronage of DST, Government of India. Research Excellence Award he has been bestowed upon for funding in 2018-19 from Faculty of Science and Technology by KBCN MU Jalgaon. He has also received Outstanding Reviewer Award from elsewhere, Netherlands. Sir has delivered many lectures as resource person in seminars and FDP, presented various papers in several conferences, published articles in international journals of repute. Sir has guided about 37 MPharm students and 5 PhD students. He also has hands-on experience on microwave synthesizer, UV, HPLC, FTIR, particle size analyzer, spectrofluorometer, and surface plasmon spectrometer, etc. He is a reviewer of some reputed journals such as Biosensors and Bioelectronics, ACS Applied Nanomaterials, Sensing and Biosensing Research, Nanomedicine and Engineering, Food and Chemical Technology, Bioorganic and Medicinal Chemistry, Current Medicinal Chemistry, Research on Chemical Intermediates. These are few of them. His major fields of scientific interest are green synthesis of graphene-based material for biosensing platform and for environmental remediation and novel pharmacophore development of several anti-cancer agents using computational tools and their evaluation against a panel of human cancer cell lines using various in vitro assay techniques. Dear participants, today he is giving a talk on graphene nanomaterials in biosensing and environmental remediation applications, the opportunities, challenges, and social relevance. Dear participants, I am also delighted to announce Dr. S.R. Lahoti Sir, Associate Professor, YB Chauhan College of Pharmacy, Aurangabad, as chairman of this session. Lahoti Sir, I welcome you, and we do expect the concluding remarks from you when the session is over.
Thank you,
so, Dr. Patil Sir, okay, welcome, sir. So, Dr. Patil Sir, we welcome you, sir. You can start your session.
Okay.
Over to you, Patil Sir.
Thank you, madam. Thanks a lot for your kind words. Am I audible, madam?
Yes, sir.
Okay. At the outset, first of all, I would like to thank Principal Dr. Respected V.K. Maurya sir for his invitation. Then coordinator of the program, Dr. Navle sir, Dr. Bothra sir, and Dr. Mahaparle sir for invitation and coordination for the presentation, questions, and everything about the session. So before starting the session, I would like to mention over here that in the morning, Dr. Saraf itself mentioned it's a time to have the interdisciplinary research. And I myself consider fortunate that I had started something different from the regular research what is going in the pharma institutions especially. So in this talk, I'm going to highlight some important part of say graphene and its implication in the pharmacy research. So let me share my screen. Is it visible?
Yes, sir.
Okay, so the topic of today's discussion is graphene nanomaterials in biosensing and environmental remediation application. So you can see from the title itself, so it's indicating something different. One material is used for the biosensing as well as environmental remediation application. So my
Slideshow is not there, sir.
slide is not visible.
Slide is not visible, sir.
The presentation is not visible, sir. Presentation.
Just a minute.
Yes, sir. You can start the slideshow now.
Now it's clear?
Yes, sir. It's clear now.
Okay. So being a pharmacy teacher and see, as far as the participants are concerned, all are very renowned faculty members from the different institutions of India. So if any query is there, or if you have any doubt, you can ask during presentation also, or whenever we have the discussion session after this talk. So actually it was an inspiring story for me because I am basically from pharmaceutical chemistry background, and my work was there on the synthesis and evaluation for different pharmacological potential, that is heterocyclic compound. But I started work on the graphene in the year 2010, and from 2010 I am working on the graphene based material for the various applications. So I think all of you know, or might be you are aware about this term graphene. Graphene is nothing but it's a allotrope of the carbon, and it is a two-dimensional planar material, and in which the carbon atom has been joined to each other like a honeycomb crystal lattice. And it was first discovered by the two renowned scientists, Andrew Jem and his student Novosalvo. So in fact, the graphite was discovered in 1564, but later on the term graphene was introduced by these two renowned scientists. And see how fortunate this professor is. His student was also involved in his research, and they have grabbed the Nobel Prize for this groundbreaking discovery of the graphene in the year 2010. So since that discovery, if you Google it out, graphene, you will find millions of paper reported on the graphene in all the aspects of the life. It is not only restricted to say physics or chemistry, but it is not restricted to physics or chemistry, but it is having potential in all the fields. So I will let you know some of the areas of research that one can choose for the further research. Now see, there are four derivatives of the graphene which we look for, or the exploration of the research. First one is the graphene. You can see from the structure itself, it's a honeycomb-like structure consisting of only carbon. Then its next part is graphene oxide, which has been synthesized from the graphene by the oxidation. And the well-known method is a Hammer's method for synthesis of the graphene oxide from the graphene. And next, its counterpart is a reduced graphene oxide, which has been achieved by the reduction of the graphene oxide. So there are several methods reported for the synthesis of the reduced graphene oxide from the graphene oxide. Maybe chemical methods are there, and nowadays people are exploring green methods for the synthesis of the reduced graphene oxide from the graphene. And one of the derivatives of the graphene is a graphene quantum dot, which is a piece of graphene fragment which actually exhibited multicolor under the light radiation of the light. That we'll see properties of this individual material in the coming slides. Now see, this is one of the slide which has mentioned the rise of the super material. That graphene is called as a wonder material because of its properties. All the properties you can by intentionally I have put this slide over here because we'll see all these properties in the presentation also. But see, it's going to change our future and bend of our smartphone. You can see 70% lighter aircraft, graphene is involved, thinner than a sheet of paper, 200 times stronger than steel, and 100% more efficient solar power. So because of this all different application, this is considered as a wonder material. And thanks to Andrew Jem and Novosalvo for this wonderful discovery. Now, as far as our field is concerned, that is pharmacy or interdisciplinary research is concerned, so it has been explored in many applications. You can find various applications of the graphene and graphene nanomaterials. It is there in bioimaging, biosensors, antibacterial agents, photothermal therapy, drug delivery, gene delivery, and tissue engineering. So bioimaging, so if you want to have idea to explore some of the agents for the bioimaging, because already we know whatever imaging is there, lots of dyes are involved. So dyes have the toxicity aspect. So one can replace toxic dye by introduction of the graphene quantum dot, because graphene quantum dot itself is a fluorescent material and it is a biocompatible one, and toxicity issues are not there with the GQDs. Then next one with the drug delivery. So you can have various aspects, that is surface modification of either graphene or graphene quantum dot for the delivery of the drug, that is a site specific drug delivery specially. Then next is the biosensing part that I will discuss in detail in coming slides. Any difficulty up to this? If you have any question, then you can raise the question, no problem. Now, the topic of my discussion today is a biosensor and environmental remediation application. So biosensor is the sensor that integrates biological element with the physico-chemical transducer, which produces an electronic signal proportional to the single analyte, which is then conveyed to a detector. Means there are major two components of the biosensor. First one is the biological element or biorecognition element, which has been integrated with the physico-chemical transducer, so that whatever signals are produced by the physico-chemical transducer in integration with the biological element, which produces a signal, electronic signal, and that signal is proportional to the concentration of the analyte present in the biological fluid. So whatever signals has been detected by the detector, then whatever signal is there, that can be conveyed to the detector and it has been amplified, and we'll get the concentration of that analyte present in the biological fluid. Now, sensors are also there, which doesn't involve the biological analytes. So biosensors are everywhere, but these are not nanobiosensors. So you can see stethoscope is there, thermometer is there, then glucometer is there, even MRI imaging is there. But these, with the help of these instruments, we cannot detect the concentration of the analyte at the nanogram level. So therefore, these are not nanobiosensors. And why there is need to have the nanobiosensor? Because we know why nanobiosensors: low concentration of the biological biomarkers in the biological fluid. Whatever concentration, some biomarkers, many of the biomarkers in the CNS, even in the myocardial, congestive heart failure, whatever biomarkers are there, they are present in biological fluid beyond the certain limit, means more than nano level, more than microgram level, microgram or milligram. So therefore, there is need to detect those analytes in the biological fluid. You can see molecular level DNA is there, but if you go in say biological aspect, you can have the different biomarkers. For example, I would like to quote here one of the example. Say, for example, Alzheimer's disease. In this, beta amyloid protein and beta secretases are there in the cerebrospinal fluid. So if Alzheimer's is there, there is an increase in the concentration of this biomarker in the CSF of that patient. And if you consider its level in the cerebrospinal fluid, it is beyond microgram level. So therefore, the particular sensor will not work. Therefore, you need to go for the development of the nanobiosensor. So this is the reason why one should go for the nanobiosensor instead of only biosensor, which can work up to only milligram or microgram level. So next part is the component of the biosensor. Already I had mentioned there are only two major part of the biosensor, but third one, detector, is also involved. So first part is the biorecognition element. Biorecognition element. Second part is the physicochemical transducer, and third one is the detector. Now see, apart from these three elements, you can see samples are there. We should have analyte. We have to try to identify the analyte present in the particular biological fluid. So you must be aware about what you are going to detect in the biological fluid. So that is analytes of the interest, a biorecognition element or biorecognition layer, or biological element. So biological element may be enzymes, antibodies, RNA, DNA, cells, or microorganism. So using this biorecognition element, one can go for the detection, fabrication and the development of the biosensor. Of course, you will have to also Google it out, prior art, before development of the or choosing any topic for the fabrication. Next is the physicochemical transducer that we'll discuss in next slide in detail. So these are the components: biological, biorecognition element, physicochemical transducer, and detector. Now, in this slide, I would like to highlight one or two points regarding biorecognition element, how to actually identify and choose the particular biorecognition element for the research perspective. First one is the antibody. Antibody is the biorecognition element, and all of us are aware about there is a interaction between affinity of the antigen towards the antibody. Once you select a particular antibody for the biosensor, and if you have antigens are itself built in. Yes, sir. Any question? Antibody as a biorecognition element and antigen as an analyte. In the second case, enzyme as a biorecognition element, and it's a biomolecule. Whatever biomolecule have the affinity towards a particular enzyme, that particular enzyme you can choose as a biorecognition element, and directly you will go for the development of the biosensor. Third one is the say DNA. DNA is a analyte and oligonucleotide sequence depending on say adenine to thymine, cytosine to guanine, guanine to cytosine. So you have to choose the analyte and the biorecognition element. This is about the ion gated channel and the biomolecule. So this here the concept of our pharmacology will help us a lot to decide biorecognition element and the analyte of the interest for the particular research. So this is a simplified version. You can take analyte, biorecognition element, and transducer. Most important, these are only the part of the or the component of the biosensor. Interface chemistry will be required over here. By suppose if you have biorecognition element, transducer, and analyte, you will not be able to develop the biosensor because you will require say interface chemistry or the nanoparticles which can have the say different sites for the absorption of the biorecognition element on their sites. So that we'll discuss in the next slide. So in brief, we'll go through all these three components, that is biological element, physicochemical transducer, and detector. So the first component is a biological element. The component used to bind the target molecule. What do you mean by biological element? Component used to bind the target molecule. Means at the biological element, whatever analyte you have decided, it will definitely bind to the particular biological element. So it must be highly specific. It should have affinity towards the analyte of interest. stable under storage condition and immobilize. So now see various examples are mentioned over here of the biological element: microorganism, tissue cell, nucleic acid, enzyme, antibody, enzyme component, and receptor. So depending on your area of research, you can choose any biological element or biorecognition element. You can see just like it is a lock and key theory of the enzyme. Enzyme, active site, substrate get perfectly fit at the receptor. Therefore, the at the biorecognition element site, target have the affinity. So it will definitely bind to the biorecognition element. Second component is a physicochemical transducer. So actually there are several physicochemical transducer, but you can opt for the whatever facilities are available at your end, so that doesn't require any sophisticated instrument. So acts as an interface, measuring the physical change that occurs with the reaction at the bioreceptor and the analyte. So whenever there is an interaction between the bioreceptor and the analyte, then there will be change in the physical properties of the molecule, and we need to detect that physical change in the physical properties of the molecule. For example, for instance, if there is a change in the electroactive substance, electrodes are there. If there is a change in pH, semiconductor pH electrode we can choose, heat, thermistor, light, photo counter, mass change, piezoelectric device. Then in our case, as far as the surface plasmon resonance spectrometer is concerned, whenever there is an interaction between the analyte and biorecognition element, then there is a change in the refractive index of the material occur, and we can detect that change in refractive index using the SPR spectrometer. And third component is the detector, that all of us know how the detector can detect the signals from the transducer are passed to a microprocessor where they are amplified and analyzed. So here only thing is that whatever facilities are available at the individual institute, you need to look out for the facilities available, and then try to have the write the project, or you can execute any project depending on the facilities available at your end. So now see, therefore I have mentioned over here, apart from the biorecognition element and the analyte, sensor material and the interface chemistry. So here actually the lots of skills and knowledge will be required to select the interface chemistry for the, not only for the fabrication of the biosensor, but even also for the drug delivery aspect. If you want to choose explore the graphene in the drug delivery, then in that sense also the interface chemistry will be required. And what is interface chemistry, that we'll see as far as my research is concerned. I don't have any other idea about the interface chemistry of the other interfering say other nanomaterials. So that we'll also discuss biological elements and analytes. So here interface chemistry will play important role in deciding your project and execution of your project at the individual institute. Now biosensor, see the types or classification of the biosensor depending on the bioelements. It may be molecular, it may be cell-based biosensor, it may be tissue-based biosensor. So in this aspect, see molecular, antibody-mediated biosensor, enzyme-mediated biosensor, and nucleic acid-mediated biosensor. Transducer, it may be optical, it may be mechanical, it may be electrochemical. So depending on this principle of operation and the instrument or the facilities available at your end, you can choose the transducer. So if you have fluorescence spectrophotometer, you may go for fluorescence detection, fluorescence based biosensor. If you have surface plasmon spectrometer, then SPR based biosensor. Absorbance UV, then electrochemical. So in this case, cyclic voltammetry will be required for, and it's, if you Google out, the major part of the biosensor development has been grasped by this development of the electrochemical and optical biosensors. So these are the few principles of operation for the fabrication of the biosensor. I am specially working on these two only, that is a fluorescence based biosensor and a surface plasmon resonance based biosensor for the detection of the biomarker in the biological fluid for early detection of the either cancer or say Alzheimer's disease. So principle of detection, if there is change in mass, piezoelectric devices, electrochemical, that is change in the electrical distribution, optical, change in the light intensity, calorimetry, change in the heat, and change in the refractive index, that is SPR, that is surface plasmon spectrometer. Now see, as far as how actually detection of the analyte is occur using the biorecognition element and say your transducer. So principle of detection is binding of the analyte into the biorecognition element, immobilized on the suitable support medium. Immobilized on the suitable support medium is refers to here interface chemistry. So it is immobilized on suitable support medium. Then whenever there is an interaction between the biorecognition element and your analyte of interest, that resulted in the change in the one of the physicochemical properties, that resulted into the change in the one of the physicochemical properties. So that I mentioned in the previous slide itself. It may be pH, electron transport, mass change, Ri, and uptake or release of the gases, or some specific ions may be also released, and which are detected and may be measured by the transducer. So this is actually the working principle of detection of the analyte using the biosensor. So usual aim is to produce electronic. So what should be in your mind? Whatever, try to identify the concentration of the analyte present in the biological fluid. So if it is reported, and then whether you are in a position to detect that particular analyte in a biological fluid using the say UV spectrophotometer or other techniques available at your end. If not, then you may go for the other detection techniques. So therefore aim is to produce electronic signal which is proportional in magnitude or frequency of the concentration of specific analyte or a group of analyte to which the biosensing element bind. So if concentration, you know the literature, whatever till date methods reported, which type of the nanomaterial has been explored for the detection of that analyte, and try to have the try to generate the novel idea out of that. And if that idea is novel, then there will be a very good output as far as your projects are concerned, or you may get very good funding from the different agencies. Even in the drug delivery, I am trying to always highlight this aspect in the drug delivery, because actually it is an interdisciplinary work, as being background from the synthetic organic chemistry or medicinal chemistry. When I started this work, actually one of my colleague, Dr. Deshmukh sir, Prashant Deshmukh, he inspired me to actually, you have the quite background in the chemistry and some of the spectroscopy part. So you may excel in this particular domain. And once I started in the year 2011-12, so till date, I am working on the graphene, and there is no any deviation from the graphene from now onwards, because along with the graphene, you can have the composite of many nanomaterials, so that there will be very good say selectivity and sensitivity as far as my topic is concerned. Now see, this slide will tell you everything about the nanobiosensor research in the graphene. So it's not only about nanobiosensor. You can just, the slide is a self-explanatory. No need to have highlight all the aspect, but you can see medical, diagnosis is there, detection, pathogen detection, pharmaceutical, even tissue engineering, fabrication of the medical devices, even stent, homeland and security, air, food and water quality, energy, health and safety. Now you can see materials, nanotechnology, fabrication, biological construction, though various aspects are mentioned over here, but frankly speaking, if you are hunt for a good research, or if you want to explore something new as out of your research, whatever current research is going on, definitely you can implement this piece of graphene in your research, and it doesn't require any sophisticated instrument or the lab setup. A preliminary characterization you can have at your institute itself. For other aspect, we can hire the different services. So why graphene is chosen? So this is actually the repetition of the same slide, which already I had mentioned at the slide number 3 or 4. But here, some aspects are mentioned over here along with its, say, up to what, say, for example, high surface area, 2630. Then strength, electron mobility, atomic thickness, transparency, thermal conductivity, electrical resistivity, and stiffness. So all these properties makes this particular wonder material, say, explore this material into the research. Now see, when I was Google out, I was came across wonderful figures. Till date, more than 31,000 patents are there in public domain, and you won't believe, out of 31,000 patent, 60%, 67% was a share grabbed by the China. Then next to China is a South Korea, around 3,200 patent. Then next to South Korea, it is USA, around 3,000 patents are there. Okay. So it's time for us to explore this material and go for the novel work. And you can also patent if you execute a novel work, and definitely it will contribute to our individual development and development of our nation also. So next is the graphene based biosensor. So majorly I would like to highlight here 3 biosensor based on the graphene. First one is the graphene based layer by layer biosensor. Second one is the graphene based antibody mediated biosensor, and third one is the graphene quantum dot based biosensor. So see, LBL is actually maybe some of our colleague working on the layer by layer application in the drug delivery. So they can also choose this particular topic. So layer by layer cell assembly is a cell assembly attractive technique for the deposition of the multi-layer molecular nanostructure, that is deposition of charged molecule and nano object depending on the area of interest or the whatever literature you have. You can choose either cation and anion, cationic polymer and anionic polymer for the development of the LBL assembly. So layer by layer deposition can provide 2 areas, solution to 2 areas. First one is the surface modification. So engineering the interaction of the given object with its environment. And third one is the fabrication of the thin film. So you can fabricate the thin film also using the layer by layer technique or layer by layer cell assembly. In fact, it is also one of the widely explored technique for the fabrication of the biosensor or the fabrication of the thin film in the various applications. So this is a schematic representation of the LBL deposition process. You can see, suppose if you have substrate, in most of the cases, I came across literature in the electrochemical detection, that is using cyclic voltammetric and amperometric detection. In that case, that electrode has been modified by the layer by layer approach. Layer by layer approach, depending on your interest, you can have many layers on the electrode, and only thing is that a final layer should it must contain your analyte of interest, so that there will be affinity with the biorecognition element and you can detect the signal. So this is your substrate. You have to coat it with the polyanion. Then it is through several washing, you can detect whether the charge is there on the particular substrate or not. We are using the potential, that is zeta potential analysis. Once the charge of your interest is there on the substrate, you can move towards the next coating of, suppose there is a anionic charge, then you have to go for the next layer using the cation. So depending on whatever layer you want, you can repeat it repeatedly. You have to repeat the procedure, but every time you need to check the charge on the surface. And if the charge on the surface is of your interest, then you can proceed for the further work. So this is actually the basic for the deposition of the layer by layer on the biosensing substrate. So it can be built through various methods. It may be electrostatic interaction, it may be hydrogen bonding, it may be step-by-step reaction, or a molecular recognition and biorecognition element. So which mechanism is involved in the LBL assembly, you need to identify, or you need to work out for whenever you confirm your analyte. Then after confirming analyte, you try to design the layer by layer assembly. How to go for the layer by layer assembly, which polymer, cationic polymer we have to use, which anionic polymer we have to use. Of course, we should go for the biodegradable polymers so that there will be no toxicity issue with the assembly. Now see, why LBL? So that nothing will be required expensive. LBL is extremely cheap and no expensive instrument is required for the fabrication. The fabrication process is simple and can achieve either manually or by machine. LBL can incorporate different functional group into the system for different applications. For example, I would like to let you know for my project how I executed that particular LBL assembly for the fabrication. Then next is the water soluble biomaterials that can be also explored in the fabrication with charge sites on their surfaces and can also be incorporated, such as proteins, DNA, and viruses. So therefore, because of these all advantages, one can explore this LBL assembly not only in the fabrication of the biosensor, but also other aspects of the drug delivery. And in fact, many of the researcher from the pharmacy background itself has explored this particular technique for the drug delivery. Now, as far as my research work is concerned, I am working on the antigen antibody mediated biosensor. First one is using the graphene, and other aspect using the graphene quantum dot. So in this case, substrate, either it may be other electrode also. So in our case, gold electrode is there. Though you need to go for the say modification of the gold electrode using the thiol group, that is thiol chemistry. And once the thiol group has been layer on the gold electrode, you need to go for the interface chemistry. So the high specificity between an antibody and antigen can be utilized in this type of sensor. About this fabrication, we'll discuss in the next slide, how actually that electrode has been modified, how the antibody has been attached to the surface of the electrode, and how the antigen is having affinity, it bind with the antibody on the surface, and how there is a change in the particular physical unit of that particular interaction, and how that change is detected by the say physicochemical transducer. So this is the actually this is about the say general aspects of graphene. Then a biosensor, its elements, analytes of interest, which are the techniques used for the fabrication of that sensor. Now here I am going to briefly discuss my project regarding fabrication of the biosensor for the detection of the Alzheimer's disease, and another is graphene quantum dot based biosensor for lung cancer. But it is not restricted, say, to my project only. Though I am exploring my work over here, but I'm damn sure about that, or I am confident about that. Whatever work I am going to discuss over here, that will be definitely useful for your further research. So it is not restricted to my work only. My work means only, say, biomarker and disease. Biomarker and disease is only specific to my work, but you can explore the same phenomenon for other aspect also. Therefore, first one is the synthesis of the metal nanoparticle AD-AB conjugate using LBL approach. So you can fabricate the uh, metal nanoparticle and antibody conjugate using the layer by layer approach. So in this case, first one is the synthesis of the metal nanoparticles. So first step we have executed is the synthesis of the metal nanoparticle. It may be silver nanoparticle, gold nanoparticle, or platinum nanoparticle. Rather, we had explored green methods for the synthesis of the nanoparticle using either microbial culture or the plant extract. So see, I am taking example over here as a silver nanoparticle. So there is a negative charge on the surface of, say, nanoparticle, silver nanoparticle. So what idea was there in my mind? I have to first coat that silver nanoparticle on the gold surface. First coating of the gold electrode with the silver nanoparticle, then identify the charge on the particular that nanoparticle. If that charge is as per my expectation, then I have to go for the next layer of another suppose anion was there, so I may go for the cation. So if in your case, if there is a negative charge, then you can definitely go for the positively charged polymer. So then I have to coat the next layer of the cation anionic polymer depending on layer of your interest. If you want 2-layer, 3-layer, then fabricate that particular polymer or the sensor gold electrode with the desired cation or anion, say polymer. Then next step is the adsorption of the antibody on the MNP, AD and AB conjugate. So the question over here is why there is no need to go for the nanoparticles. Okay. So if you go for the layer by layer assembly and maximum layers are there, then the whatever signals will get in the physicochemical transducer system, in simple word, it will enhance the sensitivity of the particular your biosensor towards detection of the analyte. That is the reason why either maybe you may go for the metal nanoparticle itself. Using metal nanoparticle, the people have also detected the analytes present in the biological fluid, but that is not too much sensitive as far as our biomarkers are concerned in the biological fluids. For example, cancer biomarkers present in the biological fluid, they are present in a very low concentration, even in the case of Alzheimer's also. So the last step will be the adsorption of the antibody onto the surface of this LBL assembly, onto the surface of the LBL assembly. So the final step will be the adsorption of your analyte. In this case, in my project, it is antibody, but in your case, the analyte may be different. But keep in mind, there should be analyte should be there at the surface, whatever the LBL assembly you have. So I think any difficulty in this case? Madam, shall I proceed next? No problem. Yes. Is it okay, na? No problem?
Yes, sir. Yes, sir. It's okay, sir.
Okay, okay. Now step second is fabrication of surface decorated graphene oxide nanocomposite for the label prognosis of the Alzheimer disease. So in fact, the idea was, which was generated in my mind, the Alzheimer's disease, whatever biomarkers are involved, so that is not actually detected at par using whatever methods are available in the literature. So therefore that idea was generated, and I was executed the write up the project, and right now it is in process execution. Say in the step second, first step is the modification of the gold electrode. Gold electrode here mentioned refers to actually whenever we work on the SPR, that is surface plasmon resonance spectrometer, actually it is with the electrode, gold electrode. So whatever the biorecognition elements are there, that we need to coat it or we need to inject it with the electrode surface. So that electrode we have to use for the further processing of our work, and that is available, means it is provided with the manufacturer itself, without just like whenever we go for the absorbance measurements, without cuvette we cannot proceed. Therefore in this particular SPR biosensor, the electrode is must, and it is a modification of the gold electrode. So that electrode has been again modified using the thiol chemistry, and SH group should be there, because that will have the affinity, or it can bind this interfering nanocomposite to the surface efficiently. So the next part is here decomposition of the GO. So reason here, exploration of the graphene oxide into the particular project. First one is it's a biocompatible one. On its surface there are abundant functional group present, that is carboxylic group. We can modify that carboxylic group using carbidamide chemistry, that is ADC NHS chemistry. And that modification of the graphene oxide using this carbidamide chemistry, though the chemistry mentioned over here, so it doesn't require say again sophisticated skill or say instrument to execute this particular carbidamide chemistry. So once this graphene oxide has been modified using this carbidamide chemistry, so you will have the amide functional group on the graphene surface instead of the carboxylic group, and that amide functionality will provide the sites for the adsorption of MNP-AD antibody conjugate on the modified electrode. Now see, first step is a modification of the gold electrode, then the adsorption, that is deposition of the graphene oxide onto the gold electrode, and its activation, actually activation of the graphene oxide on the gold electrode using carbidamide chemistry, and after that adsorption of MNP-AD antibody conjugate, which actually we have discussed in the previous slide, this one. Synthesis of MNP-ADAB conjugate using LBL approach. Whatever this conjugate we have we had prepared, we need to use that conjugate to adsorb on this GO modified gold electrode, so that that antibody will be deposited on the surface of the graphene, which is actually coated on the gold electrode, and finally there will be antibody available on the surface, antibody of our interest available on the surface of the gold electrode. And see, apart from the your expected antibody site, other sites are also there on the particular surface. So you need to block that unwanted blocking antibody sites using the bovine serum albumin BSS treatment. Once the BSA treated with this particular gold electrode, means this particular conjugate on the electrode, only the antibody of our interest will be available on the electrode. Okay. And once the antibody of our interest available at the electrode, finally there will be say interaction between the antibody is there, then you need to choose the analyte of interest, that is antigen of that antibody. So in my case, it was the beta amyloid antibody on the surface. So we have the beta amyloid antigen. Even the next step is execution of this preclinical, that is induction of the Alzheimer's using the streptozocin induced model. After inducing the streptozocin into the animal, we'll try to isolate, CSF, and we'll try to detect the presence of that particular beta amyloid protein in the CSF. Of course, clinical collaborator will be required. So Dr. Samir Goyal and Dr. K.R. Patil sir will help in this project for the execution of the detection of the antigen in the cerebrospinal fluid in the streptozocin induced Alzheimer disease model. So in that case, what happen, whenever antibody will be available on the surface of the electrode and antigen in the cerebrospinal fluid, if a beta amyloid antigens are present, definitely there will be interaction between the beta amyloid protein and beta amyloid antibody on the surface. And whenever there is an interaction between these antigen and antibody, with the help of this SPR spectrometer, that resulted into the change in the refractive index of the material. And that refractive index, change in refractive index, we need to analyze using this SPR spectrometer. Definitely there will be lots of steps. You will have to go for the calibration curve of the particular analyte and try to get the calibration curve and determination of the unknown concentration of the analyte present in the biological fluid. And definitely we have got the very good result as far as this project is concerned. And the next project is, that is concentration of the lung cancer biomarker in the detection using the graphene quantum dot and graphene based nanocomposite. So once the required layer is achieved, adsorption is completed, and definitely at every step you need to analyze using different spectroscopic technique. All the LBL assemblies, then fabric adsorption of the antibody onto the surface of the LBL assembly, deposition of the graphene on the gold electrode, modification of the graphene using this particular chemistry, whether adsorption has been occur or not of that antibody. And finally, if there is an interaction, definitely there will be change in the physicochemical properties of that particular analyte, or say physicochemical transducer. In our case, there is a change in the RI, and that we can sense the change in the RI by using SPR spectrometer. And definitely we can detect, or one can detect, the concentration of the analyte present in not only in the spike sample but also in the biological fluid. So what actually challenge I was facing since last say 2 years regarding this execution of the project. One time clinical collaborator was required, so that we fortunately we got very good pharmacology people from our end. Then in other aspect, in case of cancer, clinical aspects, whenever we go for the detection of that particular biomarker in cancer patient, so in that case still we are not able to finding some clinical collaborator. So only thing is that in spike serum sample, we are fortunate enough to analyze the say biomarker up to the say whatever the spike sample has been, spike analyte was there in the serum, and we have detected that almost about with a recovery of about 97 to 98%. So this is about the first aspect. So what idea one should get from this is how to choose the LBL, then deposition of the, or say interface chemistry. You may go for gold nanoparticle, silver nanoparticle, platinum nanoparticle, then graphene oxide is there, even silica nanoparticles are also there, carbon dots are also there, then graphene composites are there, that is graphene polymer composites also one can explore for the detection. Now next is the regarding graphene quantum dot. So actually it is a graphene quantum dot are a small nano fragment which exhibited its dimension is around less than 100 nanometer. But the what is the beauty of this particular compound or graphene quantum dot is they are emitting colors. Okay, they are self fluorescent. Okay, inherent fluorescent properties, multicolor fluorescents are there depending on the uh, graphene quantum dot you have chosen for the precursor material. Depending on precursor, some precursor are there, naturally occurring precursor, they can not only give the graphene quantum dot synthesis, but they also dope the heteroatom present in the nature, that is nitrogen, sulfur. It can easily dope with the, if nitrogen source are there, sulfur source are there in your material, then after analyzing through by using the various spectroscopic technique, we came to know that not only the carbon but other heteroatoms are also present in the particular quantum dot. So we have later on extensively evaluated those quantum dot, and with the help of EDX and XPS, we found those heteroatom has been doped onto the graphene quantum dot. So it's not only about synthesis of the carbon dot, but functionalized carbon dot. Once the heteroatoms are doped on the graphene, either graphene or graphene quantum dot, we can call them as a functionalized material, that is functionalized graphene nanocomposite or functionalized graphene quantum dot. So it represent a single layer to 10 layers of graphene or less than 10 100 nanometer, and which is physically stable, have large surface to mass ratio, and can be dispersed in water easily due to the functional group at the edges. Because of the abundant functional group present on the surface of this particular material, and hydrogen bonding can occur, and that resulted into the water dispersibility of this particular compound. So you can see the different emitted color by the graphene quantum dot depending on the precursor. So you can one can explore this particular fragment for your research independent of your area of research. So this is another aspect, again the same story over here, but the precursor material is different. Here the actually functionalization was done on the graphene of the graphene quantum dot, and which is for the early detection of the lung cancer and the bioimaging. So why bioimaging? Because of the fluorescent inherent fluorescent properties of the GQDs, one can explore this particular material for drug targeting and imaging itself. You doesn't need to go for the imaging using the dyes, as this material have the inherent fluorescent properties. Maximum researcher throughout world had explored this particular material for cancer drug targeting and imaging of the cancer in the particular organ. So synthesis of the GQD was the first aspect. Second is the synthesis of the functionalized GQDs. Third one is the fabrication of antibody, FGQD, nanoprobe for lung cancer sensing, biosensing of lung cancer biomarker in the spike sample. In this case, it is a spike sample because we till date not found any collaborator for the biosensing of this biomarker in the lung cancer. In vitro cytotoxicity and fluorescence imaging, we have got a very excellent result in these all three: cytotoxicity as well as imaging and cellular uptake. So first aspects is the synthesis of the graphene quantum dot. Actually, well-known worldwide, well accepted process is there in the literature, and that is say citric acid, carbonization of the citric acid for the synthesis of the graphene quantum dot. So it is well organized, reported, and excellent method, and maximum people are trying to explore this method only for the synthesis of the GQDs. But initially 4 or 5 years back we have approached this method, but since last 3 to 4 years we had actually explored different natural precursor for the synthesis of the graphene quantum dot, and in fact it is waste material, and we have synthesized almost explore more than 10 to 15 precursor till date, and now maximum work on the way to have good publication using, say, exploration of the natural precursor using graphene quantum dot methods. Either it may be using simple heat, then microwave assisted synthesis, hydrothermal method, and some people are explored nowadays. I came across phenton oxidation method for the high yield synthesis of the graphene quantum dot. But we are exploring only microwave means till date we have explored only microwave assisted synthesis, furnace, that is carbonization, and the hydrothermal method for the synthesis of the graphene quantum dot. Of course, after synthesis, we need to characterize them all. You involve UV, FTIR, then fluorescence, then EDX, SEM, TEM, and say XPS analysis. Now, if already graphene quantums are there, why there is need to go for the functionalization? There may be in any minds, I mean, lot of people mind there will be question why there is a functionalization. So I came across many literature, and the literature revealed that graphene quantum dot, that is bare quantum graphene quantum dot, uh, exhibited less fluorescence than the functionalized GQD. Stability of fluorescence of the functionalized GQD is more than that of the GQD. Then toxicity, actually it is a biocompatible one. So if you compare biocompatibility of the GQDs and functionalized GQDs exhibited more biocompatibility than GQDs. And this is as per not only literature, but also we have synthesized our own GQDs and functionalized GQDs. Madam, if is there any disturbance from my end, then let me know, because power shutdown is there at my end.
No, sir, nothing is
Okay.
You are very clear.
Sir, if it happens, then please let me know.
Oh, yes, sir. I will call you, or I will let you know here.
Okay. No problem.
Yes, sir.
So biosensing of the synthesis of functionalized GQDs. So we have till date explore heteroatom doping of the graphene quantum dot, that is doping of the nitrogen and sulfur, or sometime dual doping. For example, if you use thiourea, then there will be the doping of the nitrogen and sulfur simultaneously. And if you use urea, this is for an example only. Urea, one can only dope nitrogen. But we have explored till date again the natural precursor for the doping. Then apart from the doping, next aspect is the surface functionalization of the materials or the functional group which are present at the surface of the graphene, and surface functionalization using polyethylene imine and say polylysine. We explore only polyethylene imine in our case. So we have got a very excellent result as far as the if we compare graphene quantum dot and functionalized graphene quantum dot in all the aspects. In the case of sensing, in case of cytotoxicity also, we send sample for of the GQD and functionalized GQD, even fluorescence imaging. In all cases, whatever advantages are as mentioned in the literature, actually we have got those advantage with our say functionalized material. So after functionalization, there will be fabrication of the FGQDs with the lung cancer. In this case also, in our project, what I was found 3 years back, whenever I go on through literature, it was found that only CEA, the CEA antigen is was explored by many researchers worldwide for the detection of not only lung cancer, for other cancer also. But in literature, I was came across with the few biomarkers which was already reported in literature and expressed in the lung cancer. Out of that, one was the HTRD, that is human telomerase reverse transcriptase, STERT. And when that literature was found regarding expression, then I was trying to search out for the various methods for the detection of that particular biomarker. But 4 years before 4 year back, I was came across only two literature till date. At that time, only two reports were there in the public domain, and fortunately both the reports from the JNU Delhi. But that researcher has explored graphene was there, but electrochemical sensing was there. So, and only graphene was explored. But till date, again, the 3 years, almost it has a span of 2.5 years, but till date, not a single report was found on the this particular STRT sensing using the fluorescence biomarker, fluorescence sensing of the STRT. So that STRT was means conjugated with the say fabricated functionalized functionalized GQDs. So that FGQDs was conjugated with the STRT antibody, and finally that STRT antibody was there on the surface of that conjugate. And last step was biosensing of the lung cancer biomarker. Of course, the again one of the point I would like to mention over here is that cost involved. Only thing is that once the definitely the project will be good or outcome will be good, but as far as my work experience is concerned, antigen and antibody cost is too much. So another approach is there. One can choose aptamer instead of the antigen antibody for the biosensing approach. So that is my next concern. I will like to try to explore those aptamer based biosensing for the biosensing of various biomarkers. So that fabricated composite will show say STRT antibody on the surface, and whenever we take that antigen of that antibody, different dilutions different dilutions of those antibody, whenever we combine, try to combine or treat this particular antibody conjugate with the antigen, and then what we found is a quenching of the fluorescence. And that quenching of fluorescence is again there is lots of execution of work will be required. How much concentration of that antigen will quench that antibody available on the surface? Again, we need to go for the calibration curve, then different dilution and unknown concentration of the particular biomarker in the either spike sample. In this case, we have only expected the spike sample, in vitro cytotoxicity and fluorescence imaging. So in fluorescence imaging, then there is no need to go for the dyeing, and we have got the excellent imaging. Even we have got the imaging in the nucleus also. So one can target GQD for the nucleus targeting drug delivery system also. And cellular uptake, we have got the very good cellular uptake in the lung cancer cell line. So this is about the in brief about the graphene quantum dot based sensor. So here points to consider here is GQDs, exploration of the GQDs. If you don't have any idea about synthesis, then one can explore the well reported method of citric acid carbonization. Go for functionalization if you need it. If not, then plain plain GQDs are also okay, no problem. And try to execute your plan according to the reported literature. That will be not matching with exactly steps matching with exactly my talk, but you can execute the project or you can plan the project according to your interest and whatever literature available in the public domain. So this is about the glutathione biosensing. So in this case, we have achieved the nitrogen dope GQDs. Then whenever the ferric ion was added, nitrogen dope GQDs having very good exhibited very good fluorescence. When this Fe3 ion was added, then the fluorescence of the GQD was quenched by this ferric ion, and whenever the GSH was added, then fluorescence was regenerated. So depending on that concept, we need to, means we are able to detect the concentration of the glutathione or other biomarkers in the biological fluids or biological samples. Future applications, if you want to explore this particular domain, either in drug delivery or sensing, then nanobiosensor play important role for early detection of the cancer in the body fluid. So this is the most important step, identification of the biomarker. Suppose biomarker is there, lot of literature is there, but doesn't need, it doesn't write that already reported in literature, and you cannot modify that particular one. Even in reported literature, if you have a novel plan or novel composite that which is till date not reported for that particular work, then you can modify that particular nano materials and you can execute the same project. Identification of the biomarkers, validation of the cancer biomarkers, whatever literature are there, you need to just confirm those biomarkers, what are the concentration, or whether that concentration will it be able to detect those concentration using the available resources, ligands and probe development, that is interface chemistry, depending on the whatever ideas you have. Suppose you want to explore metal nanoparticle, then quantum dot, then your nano composite, polymer composite, you can execute, no problem. Cancer diagnostic biomarker detector, that is a physiochemical, choosing a physiochemical transducer for the same. Point of care cancer diagnostic, this is another aspect many people, rather in my next future perspective also I am concentrating on that point of care cancer diagnostic. So one can detect using say like a glucometer or say pregnancy strip, one can be able to detect the biomarkers in the biological fluid. Now what are the challenges? One already I had mentioned clinical collaborator and collaborations. In fact, outlook of GQDs, research on GQDs is still at an early stage. The low production yield. Production yield of the synthesis was very low till date. Up to 15% of the GQD synthesis was reported in literature. But if you go for the modification of the several methods, modification using several methods, in which I had mentioned one of the method, that is penton oxidation method, and another one is that is using coal. It is reported in literature, they have mentioned itself, but still there is need to execute or plan different methods for the synthesis of the quantum dot and try to improve the yield of the quantum dot, that is graphene quantum dot. And next hurdle is, or the next challenge is, to meet the industry requirement. Definitely it is necessary to mass production of the GQDs. In fact, there are more than 100 companies throughout world which has explored the synthesis of the graphene, but till date there is no report on the mass production of the graphene quantum dot at a relatively low cost. Definitely it should be affordable to everyone, otherwise the research will not be strengthened if you that particular material is available for the high cost. But believe me, we haven't purchased any graphene oxide or graphene quantum dot since last 10 years. We have only graphite, and which we also got from the different agency as a gift sample, and from that graphite itself we have started our work, and we are executing or synthesizing graphene oxide, reduced graphene oxide, and graphene quantum dot at our college only. Low quantum yield, that is actually the ratio of the photon emitted and photon absorbed. So if there is a high quantum yield, then there will be, definitely it is resulted into the say selective and sensitive sensitive development of the fluorescent biosensor. QIs of the GQDs, as a quantum yield is less, then I have already given you or mentioned already how to increase the quantum yield of the graphene quantum dot, and it can be enhanced by the surface modification. So in our case also, we have found a very good quantum yield as compared to synthesis of the GQDs. So these are the few challenges, but still we have the chance to look for different modification of this particular material. Now next is the challenges in the biosensor development. Despite of optimism for potential biosensor, their emergence from the research laboratory to the marketplace has been slow. You can see the overall scenario of India as compared to other developed countries. There is you will find there is a lapse between their development and our development. Still, we are trying our level best. So this is their emergence from the research lab to the marketplace has been slow. Even at institute, we will come up with very good ideas, and that ideas remain stagnant that particular stage. So the era, or one should, or whatever researcher are working, they should be able to try to find out the collaborator and try to explore that idea to the commercial aspect. So the obstacle to exploitation have been fundamentally related to presence of biomaterial in the biosensor, that is exploration of the interface chemistry and the sensor device. How to, once the idea is there, composite it there, but how you actually execute it into the point of care diagnosis of the disease, that is one of the part, or that is one of the challenge even in front of me also. So one need to try to find out the alternate solution for that. Maybe go for the means collaboration is the only key for that. Another major problem for the realistic mass production of biosensor has been the cost factor, which already I had mentioned, and the collaboration. So these are the, as I have pointed out, these are challenges for exploration of these materials into the biosensor, especially in drug delivery. I think only delivery of the gene, if I as far as my knowledge is concerned, one of my student, Abhijit Pandey, he was work on gene delivery, and he was try to execute that project along with the collaboration with the abstract. So that is possible, no problem, not an issue. So this is aspect about the graphene in biosensor. Now next is I would like to highlight in next 10 to 15 minutes graphene in environmental remediation and green graphene nanocomposite, that is graphene in environmental remediation. So again, it was a very inspiring story for me. In fact, I was up here 2 times before the DST and SERP for the final project presentation at New Delhi. But because of only one lacuna, not lacuna actually, say mass scale production of the composite. So that project was not sanctioned at the last stage. And when I was got this research grant on biosensor in 2018, so the process was diverted from the environmental remediation to the biomolecules, because ultimately it is deal with our field also and everyone's health. Therefore, actually I had stopped this work on environmental remediation since last 1 year, but it may be possible for other people to execute this work, and this is a very again interesting one and inspiring one, that is graphene in environmental remediation. This is green synthesis of graphene metal nanoparticle and composite. Composite means other than metal nanoparticle, some polymers are there. In our case, graphene is there. Graphene silver nanocomposite, graphene gold nanocomposite, graphene platinum nanocomposite, etc. Green synthesis of heteroatom dope graphene. Green synthesis of the aerogel. This is another wonder and interesting material. and green synthesis of the GQDs. So water contaminants, being pharmacy people, we all are quite aware about the contaminants in the environment, majorly dyes. So removal of oil is again one of the major contaminant in the water, and toxic organic contaminant like dyes, that is industrial pollutant and inorganic pollutant from the wastewater, and major challenging issue regarding its environmental cleanup. So whatever methods till date available are either adsorption, coagulation, chemical degradation, and filtration. So what I was found that low adsorption capacity, no recyclability of the material. Whatever material was reported, many reports are there in last 3 to 4 years, even rather many researcher from the environmental background working on the same, but still there is a huge scope to explore this material in pharmacy. Even some of the people are explore this material for the detection of the antibiotics in the pharma effluents, detection of the antibiotics in the pharma effluents, low adsorption capacity, poor recyclability, and these two are the major disadvantages associated with whatever methods. Need and correlation with the present scenario: leakage of crude oil and petroleum products, inorganic ion and organic pollutant, and dye contaminants, serious environmental and ecological problems. So all of you know that is leakage of the crude oil at sea surface. Some conventional methods, that is combustion oil booms and porous absorbents, were employed to address these issues. So therefore, and generally they are suffer from or exhibited low absorption capacity, poor recyclability. Therefore, I was thought to explore the graphene. Instead of graphene, reduce graphene oxide, and it exhibited the high surface area. If surface area is high, then there will be very good absorption properties to the particular nanocomposite or say aerogel. Now see, in this case, it is simple Moringa oleifera shell we have explored for the synthesis of the graphene silver nanocomposite. So in that case, what we select is shell of this Moringa oleifera, which is actually waste. It has been discarded. Then extract of that shell was prepared. Then simultaneously graphene oxide solution was prepared and silver nitrate solution was prepared. So there is a debatable topic on whether sunlight assisted synthesis of the composite was there. Definitely there is an issue with the say intensity of the sunlight, but try to choose the particular time on every day and try to repeat the same thing, either duplicate and in triplicate, so that you will get the consistent size and say size of the nanomaterial and say other evaluation parameters for the that particular composite. So what happens in this case, actually this particular extract, the idea was extract, already we know extract consists of the various phytoconstituent, and various phytoconstituent, for example, phenols are there, phenolic component, flavonoids are there, tannins are there. So maximum phytoconstituents exhibited reducing properties. So in fact, those constituent we need to identify, we need to check the phytoconstituent present in the extract, and with the help of that phytoconstituent, it may be possible to reduce in situ graphene and silver into reduced graphene oxide and silver nanoparticle. There is a formation of the silver nanoparticle and reduced graphene oxide in the single composite. And we need to detect all these, we need to detect this formation of the composite using various spectroscopic technique. In this case, it's a UV, FTIR, then your EDX and TEM. After confirmation of the fabrication of this particular composite, you need to go for the detection of either inorganic ions, dye degradation, or removal of the environment pollutant in the water. So in this case, there is a detection of the lead, chromium and lead in the using this silver graphene nanocomposite. Second case was with the actually this work was published in the research on chemical intermediate. So the requirement was catalyst should be different. So in our case, plant extract was there. So we got major and minor revision from this particular journal. So we try to produce this particular output. This was again published in Russian Journal of Physical Chemistry. So in this case, instead of the graphene silver nanocomposite, we fortunate enough to synthesize the graphene gold nanocomposite. So in this case, the precursor was a chloroauric acid and very less concentration was required using banana peel extract. Actually, it was explored in the three application. First one is the dye degradation. Second one is the catalysis. In this case, catalysis is a 4-nitrophenol, which is a toxic one. Once that conversion of the 4-nitrophenol has been with the help of course, it is a reduction reaction, that is 4-aminophenol. By using this particular composite, 4-aminophenol was achieved. Chromium detection, but we have restricted our work to this only. Again, this is using RGO AZNC is using banana peel extract, another work. This is for the say metal nanoparticle, sorry, magnetic graphene nanocomposite. Using graphene and ferrofluid, we had synthesized the graphene magnetic nanocomposite for the detection of the environmental pollutant. Now next part is, this is about the graphene composite, graphene metal nanocomposite. So in the next aspect of my talk is to heteroatom doping or the functionalization of the graphene material, that is introduction of the heteroatom, introduction of heteroatom, that is nitrogen, sulfur, phosphorus, boron into graphene sheet is a generally effective in modifying their electrical properties and chemical activities. So what happened, instead of the graphene, if you functionalize it with either say functionalizing agent or say doping material, then you may be able to detect or say enhance whatever the plan in your mind. For example, in my case, it was a degradation of the environmental pollutant and absorption of the oil from the water. So instead of the graphene, if heteroatom dope graphene was used, so report literature was revealed, after doping of the graphene with the heteroatom, it resulted into the increase in the surface area of the graphene. So once the surface area was increased, definitely it will give you the best absorption properties. So actually might be, I don't know how many of you are aware about this term aerogel. So aerogel are a class of porous material with excellent physicochemical properties such as a low density, high porosity, high surface area and adjustable surface chemistry. So again, one of the challenges I was found with this particular exploration as far as the aerogel is concerned, that is say BET surface analyzer. And if you want to characterize one sample, it is costing around more than 2,500 for one sample. So we have been again fortunate enough within next 1 or 2 months, BET surface analyzer will be available at our end, at our college. It is generated from Chattap Sir's grant. So among the various high performance applications, aerogel has drawn significant attention as an adsorption media for removal of several environmental and human health threatening pollutant. So issue was pore size and pore distribution and surface area we have to determine. But because of the cost factor, we have not estimated that for our synthesized material till date. So among the that removal of several environmental and human health threatening pollutant. So if you combine these two terms, that is graphene and aerogel, we will get excellent material, that is graphene aerogel, which can be proved as a promising material for environmental cleanup. So people had explored many material for the synthesis of the aerogel, but in our case, even graphene was also explored by many scientists, because I think this work was reported by us in the year 2017, I think. Additionally, doping will help to further tune the surface chemistry. So again, one of my student, as the facility will be available, we have allotted this work, modification of this particular material by the other aspect for the further application. Although aerogel performance for environmental remediation are promising, some drawbacks of aerogel are again there. Intricate drying process, drying is difficult, mechanically delicate structure, and the most important one is we cannot go for large scale here at our end or at laboratory scale. Low mechanical stability and potential for disposal problem. So these are the some problem associated with this graphene based aerogel or aerogel. So we have tried this particular fabrication of nitrogen and sulfur co-dope graphene aerogel using a very simple method, graphene oxide and thiourea. Thiourea again contains sulfur and nitrogen. Heat at 90 degrees Celsius for 6 hours, you will get hydrogel. And after lyophilization, you will get this particular material. And frankly speaking, this is from our lab only. That is nitrogen and sulfur co-dope graphene aerogel. And now you can see the application. That is environmental contamination, means various dyes are there. You need to prepare a specific concentration of the dye solution. Then draw the UV curve, whatever lambda max is there for the particular dye. Then you have to add the specific amount of the, again, it's on trial and error basis, of course. Try to add the minimum, say, concentration of the particular composite and try to execute how much particular concentration is required to decolorize the 100% dye. And then these are the original photos from our lab only. This is methylene blue and malachite green. And with the help of this aerogel, there is a complete decolorization of the dye. Next one is, I think it is a very, say, interesting topic for us that time, oil absorption study. Significantly, oil is float on water, everybody know. So will there be any method which can only absorb oil from the water? So we have explored this nitrogen and sulfur dope graphene aerogel for the absorption of the oil from the water. Okay. So actually oil has been treated with the Sudan red. So it appears in the red color. And wait for a minute. I will try to show you the actual how the oil has been absorbed by our synthesized aerogel. This is a water and oil has been treated with red, and then aerogel was added to the particular mixer. Okay. So in that sense, we have explored this nitrogen and sulfur dope graphene aerogel for the absorption of the oil. Now next is regarding nitrogen dope graphene magnetic aerogel. This was previous one was the wait for a minute. So in this case, graphene synthesis, nitrogen dope graphene was synthesized and it has been clubbed with the magnetic nanoparticle for the environmental remediation. So the concept was same. Instead of graphene, so the actually bone meal extract was used, and bone meal extract actually bone meal was bone meal is say rich source of the nitrogen. So therefore, because of this extract, nitrogen has been doped on the surface of the graphene, and this is not doping has been confirmed by EDX analysis as well as even elemental analysis at routine organic chemistry lab and XPS analysis. After that, it has been treated with the magnetic nanoparticle which was previously synthesized, and that nitrogen dope graphene magnetic aerogel was explored for the degradation of the dyes. So what is the beauty of this work is as because of the magnetic nature of this composite, we can use same composite for the repeated cycle. So you won't believe up to 5 cycles, this same composite was able to decolorize the dye up to 75%. So this is about nitrogen dope graphene aerogel. This is again fabrication of the NRGO for synthesis of the nitrogen dope graphene. So this is about magnetic aerogel, NRGO and magnetic nanoparticle. This is silica, nitrogen dope graphene silica aerogel. So depending on say your knowledge and literature, try to plan some novel work and try to execute the plan work. So this is final slide as a concluding remark. So as a potential threat for bioterrorism increases, there is a great need for a tool that can be quickly, reliably, accurately detect contaminating bioagents in the atmosphere. So this is again one of the booming area of research for those who are working in the biosensor. Bioweapons detection, contaminating bioagents in the atmosphere. Biosensor can essentially serve as a low cost and a highly efficient devices for this purpose, in addition to be being used in other day-to-day application. To fabricate eco-friendly and economically selective biosensor for point of care diagnosis of the disease. So if you are able to develop fabricate biosensor, which will be able to detect say biomarkers in biological fluid, say within 1 minute, 2 minutes, or say within 1 hour, then that will be also okay. The 1 hour for the say critical biomarkers. Okay. But only thing is that we need to work on say cost effective fabrication. Okay, sir. Thank you. This is from my end. Questions are always welcome. If you have any difficulty, you can ask me, and hope I will be happy if you have any query or answer a few questions.
Thank you, sir. Dear participants, the session is open for discussion. If anybody wants to ask any queries, any doubts, you can ask.
Yes, you can use other electrode, but depending on your choice. Suppose if you go for electrochemical detection, there is no need of the gold electrode. One of the question is there in the chat box.
Okay,
If you want,
chat
you
box.
can ask the question in the chat box itself.
Sir,
Yeah, ma'am.
may I ask a question?
Yeah, yeah.
Sir, actually, I'm working on a project where
Yes.
I wanted to detect the levels or the concentrations of the metabolites of our biogenic amines, like
Metabolites of?
biogenic amines,
Yeah,
like
yeah, biogenic
serotonin and
amines. Okay,
noradrenaline.
okay,
Yeah.
okay.
Okay, so homovanillic acid and vanillic
Yeah,
acid.
yeah,
that
yeah. I
I
know,
want.
no problem.
So can we use this biosensors even for the detection of that in the
Yeah,
biological
yes,
samples?
madam. Definitely, definitely, no problem. In fact, it is one of the way to track the pharmacokinetic parameter also, because along with the biological amines, there are many reports for the biosensing of dopamine, then your acetylcholine, all the biomarkers. And your work is beyond that. If you want to analyze metabolites form after metabolism of those biogenic amines, then that will be very worthy, no problem, not an issue. You can definitely
So
detect.
we can try this graphene quantum dot or whatever
Yes, yes, yes.
biosensors you
No,
have
no,
mentioned in
no
the
problem. Only thing is that as it is a metabolism, then I don't know, but the clean pharmacology people will be required.
Yes,
If you
sir.
are
I'm a pharmacology.
yeah,
I'm
yeah,
a
then no,
pharmacology
not an
person.
issue. You
Yes.
just have the solution from the biological fluid, and you can detect those biomarkers.
Thank you so much, sir. And sir, can I get your email ID here so I can
Yeah, yeah,
contact you personally and
yeah, yeah,
can
no problem.
work in collaboration?
But it will take a time. You
Right.
need to have the plan. Then
Yes, sir.
after
Actually,
planning,
I've
everything.
already I've started working on it.
Yeah.
So some standard graphs and all that thing we have already prepared in
Yeah.
the artificial samples.
Okay, okay.
Now we are trying in the actually biological samples.
No, no problem, madam.
Yes, sir. Yes.
Definitely, I will be there. No, not an issue.
Yes, sir. Thank you so much, sir. Thank you.
Yes, anybody else wants to ask any query to Patil sir? Do we see something in chat box? I think we shall declare that the session is over. So thank you, Dr. Patil sir, for enlightening us and with an altogether novel concept of using graphene nanomaterials. I now request Dr. Lahoti sir to please conclude the session.
Am I audible, ma'am?
Yes, 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 sir.
Okay. So good afternoon, one and all here. I actually, at the outset, I'm thankful to team GCOP, especially Navle sir and the team of Government College of Pharmacy for organizing such a short-term training program for the faculties in the region. And in the inauguration function itself, Dr. Saraf, Honorable Dr. Saraf sir, Dr. Maurya sir, and Dr. Venkat sir has given the emphasis on the smart drug delivery system. And because this is a time to work on this smarter basis rather than the conventional basis. So in line with that, Dr. Praveen Patil has given a very excellent view on the special topic of graphenes in the biosensing and the environmental application, challenges, and the social relevance. Till now, we are thinking that the carbon is having only one precious allotrope, that is diamond. But after this particular talk, we come to the conclusion other than the diamond, there is more precious allotrope, that is graphene. Because this, in his overall talk, he has given in-depth information regarding the graphenes, right from the synthesis of the graphene with the green method. This is a wonder material. And with this particular talk, we come to know that this is a very wonderful material which can be used into the drug delivery system. Actually, this graphene is not new. It is used into the paint and various application in electrodes and electronics and DNA sequencing. But what I come to know that this graphene is recently been, I think it is very recently used into this drug delivery system. He has given the total idea about the nanobiosensors, composition of the nanobiosensors, fabrication of the nanobiosensors. Then along with that, how this biorecognition can occur, which are the various principles of the biorecognitions. Then how to choose this biorecognition elements, then how to choose the transducers, signal generators, then how to go for detection of these biosensors, then also how this analyte can be detected, how to decide the analyte that can be detected. Then also given the idea about the biosensor techniques, fabrications like LBL and the antibody mediated techniques, quantum dot techniques. Also, he has given the also discussed which are the challenges he faced during the research work, because in academics to carry out this sort of research is a very difficult task. So he has given which are the various challenges he faced. Also, he has given the overall how it can be this role of graphene into the environmental remediation, to reduce the water contamination. Also, given the very excellent his work, how the dye can be removed from the water contamination, how the environmental contamination can be reduced by these biosensors using this graphene technique. Then also he has given the idea about the heteroatoms doping techniques, that is using this heteroatoms for synthesis of the or to modify the characteristics of the graphene. And also he has given the idea how this agro waste is very interestingly, agro waste can be used for synthesis of this nanocomposite fabrication using graphenes, and that can be used for the environment. And I think, sir, this graphene can also be useful for the targeted drug delivery system, especially for the cancer, which is thrust area. Also, it can also be useful for the certain responsive drug delivery system like pH responsive drug delivery system. That can also use thermosensitive drug delivery system, that can be very well useful. Even light sensitive drug delivery system, it can be very useful, this graphene biosensors. So with your talk, you are given a unique combination of the chemistry, pharmacy, physics, electronics, all this is a blend of interdisciplinary research, and this has given new dimensions to our research. So definitely we will try this. One query is there because of this work. So can we use, because you already answered that query during your talk, but always there is a question in mind, and सभी के दिमाग में एक क्वेश्चन होगा कि whether the facilities requirement, because for this type of research, how much of the facilities that requirement is there for the college, because maximum academic institutions, they are facing with the scarcity of the facilities. Okay. So that is questionable. So which are the minimum facilities that require for this type of research work, sir?
Yes, sir. Yes, sir. Definitely, I will let you know. Before that, thank you for your kind words. And as far as targeted drug delivery is concerned, you already mentioned. Actually, whenever you have any idea of the drug delivery, then it is better to have the stimuli. If there is any stimuli, for example, pH responsive drug delivery system, or the pH of the environment where you want to target, and if you are able to fabricate such a composite which can be degraded in that particular pH only, then that will be very excellent topic. The maximum people are trying to fabricate that drug delivery system depending on the pH of the environment using graphene itself. And as far as the facilities are concerned, frankly speaking, sir, when actually we were started in the year 2012, so we have only UV spectrophotometer and other instrument the other colleges have at the B Pharma level and PG level. UV and FTIR. Only UV and FTIR. If you have UV, if you go for the graphene quantum dot without fluorescence, you cannot move. For graphene quantum dot, you need a fluorescence spectrophotometer. But for graphene based material, except quantum dot, your simple organic synthesis setup will be required. That is magnetic stirrer, then your hot air oven, muffle furnace, UV spectrophotometer, and FTIR. So that in the initial study, if you confirm those things, whatever required for the confirmation of the composite, later on, then if you have say Zeta sizer, maximum college don't have that facility, but we haven't same facility when we started work. We have Zeta size facility after 2015 only. So 2010 to 15, we have been higher zeta size and potential facilities. Now we have, then that is not an issue, but up to this it's okay. UV, FTIR and other facilities required for the UG program. And for other
So,
characterization, we are also going for SEM, EDX. We need to hire those facilities because we don't. One point I had highlighted regarding aerogel, that is BET surface analyzer. Till that, except BET surface analyzer, whatever work was mentioned in the slide, up to 80% of the work we have facility here at our end only. Thiourea, graphene, and hot plate. Nothing beyond that. And for characterization, UV and FTIR. After that, remaining facility at our end also we need to hire. So
very
as far as means in the simple word, as far as if you want to explore this particular material in your research, facilities won't be required except hiring analytical services, because maximum college have those facilities as far as my knowledge is concerned. If you have PG program, then you don't need to worry about execution of the work.
really wonderful, sir. Actually, you have motivated us, because up till now we are thinking that for good research only the facilities are important. But from your research work, we come to know that facilities are not important. More important is your own ideas, and ideas are very, very important.
Yes,
So
yes.
thank you very much, sir, for your wonderful work, and we will be definitely.
Just I will take
Yes.
1 minute more for your kind information, or for just sake of your information only. See, I'm working from this area. From this year, we have started one more wonder material, that is metal organic framework, MOF. Conjugation of the graphene quantum dot, graphene and metal organic framework. In fact, my all the 4 PG student this year working on MOF, and 1 PhD student I have assigned him work on graphene MOF composite for the biosensing application. And we won't believe, we have published 2 papers on MOF, and 2 we have submitted revision, more than 3 impact factor. So this is also
Wonderful, sir.
one of the great area to be executed, but that will require in MOF case some pharmaceutical chemistry background will be required. Thank you, sir. Thanks a lot.
Thank you, sir. Thank you, sir. Thank you very much for your wonderful session on this graphene. And definitely all the participants, they are benefited, they are motivated to work on some different areas with the other than the conventional areas. And thankful to the Government College of Pharmacy, Navle sir, Mayura madam, and the respected Maurya sir for giving me opportunity to chair this session, wonderful session of Dr. Praveen Patil sir. Thank you very much. Over to Mayura madam, please.
Thank you, Lavati sir. First of all, let me thank you that you have made out valuable time from your busy schedule, your college and academics and everything, and be a part of our STTP. So thank you very much, Lavati sir. And now I would request Navle sir to display the certificate as a token of appreciation and love from our college to Dr. Patil sir.
Thank you, sir. Thank you, Navle sir.
I think that calls for an online applause from all of us.
Thank you, madam. Thank you, team GCOP, and all the participants for patient listening.
Thank you, sir.
To all participants also.
Yes, sir. Yes, sir. Over to you, Navle sir, if you want to give any instructions to the participants.
This is for Lavati sir.
Okay.
Certificate for Lavati sir.
Thank you, sir. Thank you.
Thank you very much, Patil sir. Thank you very much, Lavati sir, for your chairing the session. Praveen sir, your work was very excellent, really. Thank you very much for your sharings. There is one only announcement to the participants that we will sharp start our session at 2 o'clock. Very excellent speaker in afternoon we are having, Mridula Bele. So I will start this link at 1:45 sharp, and immediately we will, so you joined it at 1:45, so that we will start exactly at 2 o'clock. There is one more instruction I will say to all the participants, that please mute your mic, because there are much more disturbances to the speakers during that. Plus, stop your annotation. Someone is playing with your annotation. So there are many time interference in that. So please keep your mobile in a safer place. If you are listening from the distance, don't hand over it to your childrens and all these things. So they will play with that mobile and annotation is made on, or your mic is made on, and it is creating the disturbance to the speaker. So thank you very much. See you at 1:45 p.m. Thank you very much.
you, sir.
Thank you all.
Thank you.
Thank you.
Thank you, sir. Yeah, thank you.
It's about IPR, and I'm going to try to answer those questions which all of us have in our mind when it comes to patenting of our inventions, because as I said, filing a patent is not our day-to-day job. We are more used to writing research papers and publishing papers. So when it comes to patenting our invention, we have several questions. So just to start the session on a lighter note, these are some beliefs that we carry in our mind. We do not know whether they are true or not, but we believe in these. So there are several black holes in the space. Bananas grow on tree. We evolved from chimps. These are the beliefs we always carry throughout our life. You believe that all these facts are correct, right? Don't you? We all believe that these are the true facts. But they can be incorrect at times, and we do not always take efforts to find out the true answers to these beliefs, to try to verify these beliefs, okay? And they can be incorrect. So when it comes to intellectual property, now, having little information or no information can be very risky, especially we are talking about patents. Now, why it can be risky? If we believe that we evolved from chimps and that belief is not true, there is hardly any risk involved. But if we do not have the correct information about patents and we go for patenting, then it can be risky. Why? Because it can cost you millions of rupees in revenue. You can earn millions of rupees if your patent clicks and it gets commercialized, but it can also lead to a potential lawsuit. You can be sued, you may have to suffer punitive damages or fines, and therefore it is very important that we burst these myths which we have in our minds about patenting of our inventions. So let us today try to bust some common myths about patents. What exactly? So let's start with basics. Let's start from scratch. And what exactly is intellectual property? Okay. We are used to only two types of properties, movable and immovable properties. Especially in India, we are not concerned much about intellectual property. So what is this? What is intellectual property? As the name suggests, it refers to creations of mind or the intellect. Okay, so anything which is created by your mind and it can be commercialized, you can earn money using that, can be called as intellectual property. Intellectual property rights are the legal rights governing the use of creations of the human minds. Okay, so there are two terms which you will hear. One is IP, that is intellectual property, and the other is IPR, that is intellectual property rights. So intellectual property rights are the legal rights which govern the use of creations of the human minds. They usually give the creator an exclusive right. We'll talk about this right in detail later, but generally the one who owns the intellectual property has the exclusive right on that property to use it. He has the exclusive right over the use of his creations for a certain period of time. We should always remember that the right is limited to a certain period of time and it dies, it expires after a period of time. We know that there are several forms of intellectual property like trademarks and patents and industrial designs and geographical indicators and copyrights and patents. So patents is one of them, and we are going to talk about patents only today. Okay. Now there are certain characteristics, certain features of it. May I request everyone to mute your mics, please? Can you please mute your mics?
Excuse me, please mute.
Yeah. So we are more used to owning properties like land or gold or cash. Okay, so these can be counted, these can be measured. How many acres of land I have, or how many kgs of gold I have, I can count it. Okay, but intellectual property cannot be measured, it cannot be counted. So it is an intangible property, whereas the other forms of properties are tangible. We cannot measure intellectual property. Therefore, the first important feature is it is intangible. Secondly, it is non-rivalrous. Okay, now what do we mean by this? Now, what you're seeing here on the screen are the sentences by the famous Thomas Jefferson, who is supposed to be the father of intellectual property as far as United States of America is concerned. So he said that he who receives an idea from me receives instruction himself without lessening mine, as he who lights his taper at mine receives light without darkening me. So taper is a kind of light, ज्याला आपण मराठीत मशाल म्हणतो. So if somebody lights his taper at my taper, I have a lit taper in my hand, and somebody takes light from my taper, then my light does not get lessened. If somebody shares my light, it does not darken me. Similarly, if someone receives an idea from me, if someone uses my idea, then I still have it. I still have the information. Nobody can. So, for example, if I have 5 acres of land, okay, and somebody encroaches upon 1 acre out of 5 acres, so I am left with 4 acres. So 1 acre is reduced. The 1 acre is seized by the other person who encroached it. So I'm left with 4 acres, right? But if I have some idea and somebody uses or steals that idea from me, I still have it. It does not lessen my knowledge. It does not reduce my information. And that is why it is said that intellectual property is non-rivalrous. The other forms, we know the word rival or rivalry. So other forms of property cause rivalry. For example, I gave you an example of land, and we know what kind of disputes or what kind of rivalry a land can create, a property like land can create. But in case of intellectual property, if somebody uses my idea, I don't get deprived from that idea, and therefore it is called as non-rivalrous. The third feature of intellectual property is it is non-excludable. Again, if I have a land and I have to protect it from encroachment, I put a fence around it. You can see a fence here. Or if I have gold, I keep it in locker for saving from others. But I cannot build a fence around idea. Okay, ideas cannot be fenced, and therefore it becomes very difficult to exclude others, to stop others from using your own ideas. And therefore you need laws. For example, you need patent laws to protect your invention, to protect your ideas so that nobody steals it. Okay, so being intellectual property is intangible, it is non-rivalrous, and it is non-excludable. These are the classical distinguishing features of intellectual property, which distinguish it from the other forms of property like land and gold and money. Okay. Now, there was a very famous scientist by the name of Arrow. Okay, and we are talking about the economics of patents. So what happens is, suppose you and me, people like you and me who are academic researchers. So suppose I have invented something in my lab and I want to commercialize it. Okay, suppose it is some kind of formulation or some kind of device used for administering a drug. I have invented it and I want to commercialize. So obviously, I do not have any industry or do not have the facilities so that I can commercialize on a larger scale. So what I would do, I will approach someone from the industry who holds a similar kind of industry, and I will ask him or her whether he or she wants to buy my invention. Okay. Now, when I approach someone like this in order to commercialize my invention, I will have to disclose what my invention is. Just a moment. Yeah, so I will have to disclose my invention, and then only that person will decide whether he wants to buy my invention or not. So in order to sell my invention to him, I will have to disclose it. But then what can happen? If I disclose my invention, I tell him completely that how do I manufacture my invention, how to make my invention, then after listening to everything, after understanding each and every minute detail of my invention from me, he may say that I don't want to buy it now. Okay, and then he can start making it himself because I have disclosed the invention to him. When an academic researcher like me approaches someone in order to commercialize my invention, then I am always caught in the dilemma. Okay, he would not buy it unless I disclose it, but I am scared to disclose it because I fear that he may steal it without buying it from me. Okay, so I'm always caught in the dilemma whether to disclose my invention or not. Okay, both the things are equally bad for me. If I don't disclose it, he will not buy it. But if I disclose it, there is a danger of him stealing it. So this dilemma of whether to disclose or not is called Arrow's information paradox. Okay, so in such situation, patents become our friends, because once I file a patent and then I approach this person, then even if he does not, after disclosing my invention, he does not want to buy it, he cannot steal it because I have a patent in place. So that is a importance of patents, which stops the problem of free riders. Free riders are those who want to use the information, who want to use some goods. Patents are also called as goods. They are commercial goods. So people want to use goods without paying money for it. They are called free riders. So this presence of patent will reduce the problem of free rider and free the inventor from this information paradox, or from this dilemma of whether to disclose or not to disclose my invention. So patent laws, what is the importance of patents, or what do the patents offer to the inventor? They offer three kinds of incentive: incentive to invent, incentive to innovate, and incentive to disclose. Okay, so why should I invent? Because it may happen that if I invent something which is commercially viable, then I may be able to sell it, and I may be able to receive money in return. So I get incentivized to invent. What is the difference between invention and innovation? When an invention reaches a commercial stage and it is being manufactured on an industrial scale, then it is called as innovation. That is the difference. So invention is always limited to a lab scale, whereas innovation is an industrial scale. So in order to innovate, when I have to convert my invention to innovation, I have to approach some industry and disclose my invention. And why should I disclose? What is the incentive I get if I innovate it? I will earn money. So I get incentivized to invent, I get incentivized to innovate, and I get incentivized to disclose. In absence of patents, the inventor would not be ready to disclose his invention completely. If you have seen the research papers, many times the researchers tend to skip the important steps in the research. Okay, but if I have to earn a patent, if I have to get a patent granted for my invention, then I must disclose it. And why do I disclose? Because I know that unless I disclose my invention, I am not going to get a patent. Okay, so patents provide an incentive to invent, an incentive to innovate, an incentive to disclose the invention to an inventor. Okay, so what is a patent? Basically, patent was derived from the word literae patens, which actually mean open letters. So this is an example. This is how a patent looks. You must have seen several patents. So this is a cover page disclosing the bibliography of the patent. These are the diagrams, abstract. This is called bibliography, which tells you the title and who are the inventors and assignee and application date and so on. Okay. So what is a patent basically? Patent is a right which is issued by the government. Patent is always issued by the government. So patent is a government issued right, issued grant conferring that the owner of the patent has the right to exclude others. The owner of the patent has the right to stop others from making, using, offering for sale the claimed invention, but the patent does not give its owner a right to use the claimed invention. You may be surprised to hear this. We'll talk about this later. So patents are always said to be a negative right. It is a right to stop others from using your own invention. Okay, now another buzz. So this was about the preliminary introduction to IP and patents. First myth which we always have, I have seen many academic researchers saying that I won't be able to get a patent because I am a small academic researcher. I am not developing any rocket science. I am not developing any breakthrough idea. So it is impossible that I can get a patent on my idea. No, this is a myth. In order to get a patent, it is not necessary that your idea has to be a breakthrough idea. Okay. In fact, many times the researchers or the patented ideas are always what we call them as incremental inventions. An incremental invention is there a lot of disturbance from my side? Are you getting disturbed because of this background noise?
Yes, but it seems that no other person is doing it because I'm just observing it and everybody is muted.
No, it's from my side only. Is it very disturbing? I'll have to.
No, ma'am. Please continue, ma'am.
It's bearable.
It's bearable.
Some construction work is going on outside.
No problem.
Yeah. Okay. So it need not be a breakthrough research. Most of the times, the patented inventions are incremental inventions. So just an extension of already existing invention. Just to give you an example, I have played some funny ideas which have been patented. What you see here is a bird diaper. Okay, so this is a patented product. Or you might have heard about an Indian school girl earning a patent for these type of stapler pins. So what happens is when we are using the staplers, at one point of time, the pins get over and then the stapler stucks. Okay, it gets entangled. So in order to do that, in order to solve this problem, what that girl did was the last two pins in the bunch of these pins, stapler pins, she colored them. So if you get a first colored pin while you were stapling, it is an indicator to you, it is an alarm to you to indicate that the pins are about to get over and you should refill it. So such kind of small inventions can also earn a patent, and it is absolutely not required that you have to have a breakthrough idea in order to get a patent. Then, as I mentioned before, patents are negative rights. So we always believe that once you get a patent, you can exploit the product in any which way. Once I have got a patent on a particular, for example, I am an academic researcher and I have developed a formulation for which I have got a patent. So I always believe that because I have got a patent, now I can anytime start manufacturing that formulation. Is it true? No, it is a wrong belief. And patent, as I said, is a negative right. It does not always give the owner the right to exploit your own invention, but it always gives you a right to stop others from using it. So patent is a negative right and it is not a positive privilege. The patentee has a right to prevent the manufacture, sale, use, importation, or offer for sale of the patented product, process, or composition. But it is not a positive right, and it may not enable its owner to do anything that he or she wants. Why am I saying this? This statement might have confused you. So let us see an example. a very simple example. So this is a person named Sharad, and he has a patent. So Nita has a patent on a simple lead pencil. You can see here a simple pencil. And Sharad has a patent on a pencil with an eraser. Okay. So one person has a patent on pencil, simple pencil, and the other person has a patent on. So you can see here, this is an incremental invention. Nita had already invented a pencil. Sharad added an eraser to this pencil and he got a patent on this. Okay, so Nita has a patent on plain pencil and Sharad has a patent on pencil with an eraser. Okay, now can Sharad sell a pencil without an eraser? Once he gets a patent on this, can he sell this pencil? Or can Nita sell this pencil? Or basically, can Sharad make this pencil himself? He has a patent on a pencil with an eraser. So if can he start making this pencil? Probably yes, probably no. Why not? Because when he starts making this pencil, first of all, what he is going to do, he is going to make a plain pencil and then he is going to attach a eraser to it. Okay. So when he makes this plain pencil, Nita has a patent on this plain pencil. So if he wants to make it, he is infringing on Nita's patent and Nita can stop him from making it. Though Sharad has a patent on a pencil with an eraser, he may not be able to make this pencil without Nita's permission. Now, can Nita make this pencil? Probably not, because if she tries to make this pencil, she will infringe on Sharad's patent. So in this situation, Sharad has a right to stop Nita from making this pencil. Nita has a right to stop Sharad from making this pencil, but they may not have the rights to make their own patented pencils. And that is what I mean when I say that patent is a negative right. It may not give the owner the right to exploit his invention. So in such situation, what is happening? These type of patents are called blocking patents. So Nita's patent is blocking Sharad from exploiting it, and Sharad's patent is blocking Nita from exploiting it. So what do people do in such situations? They will have to enter into a cross licensing agreement, which will say, Sharad will say that you can make these pencils without my permission. I allow you to make these pencils, but you will not make my type of pencil. And I will only make this type of pencil. I will not make your type of pencil. But in order to be able to make their own pencils, they can simply not start making these pencils. They will have to enter into a cross-licensing agreement. They will have to give royalties to each other, and then they can start making this. That is what I meant when I say that patents are negative rights and they are not positive privileges. That is what has been explained in this slide. So I skip this. So it's like, for example, it's like owning a car. So if you buy a car, you have purchased this car, you have got the keys. It does not give you the right to drive it. Okay, unless and until you appear for the RTO exam and you get the driver's license, you will not be able to. There are certain other conditions, like you need to have a license, you need to have a number plate, insurance, etc. So owning a car does not give you the right to drive it. Owning a car does, however, once you own a car, you always have the right to stop other from driving your car. But you yourself may not be able to drive it unless you fulfill these other conditions. So it is exactly similar to patents, which are supposed to be a negative right. It is not a positive right that enables its owners to do anything that he or she wants. Okay, so this was one question. Then the second myth which we have, that I will file a global patent on my invention and then my invention will get protected throughout the world. Is it true? Can I file a global patent on my invention? No. There does not exist something called as global patents. Global patents hote hi nahi hai. Okay, why? Because patents are territorial rights. So if I file a patent in India and I have got a patent in India, then my invention is patented only within the geographical jurisdiction of Indian Patent Office. My patent is protected only within India, within the territory of India. So if somebody infringes on my patent in the United States of America and where I have not got a patent, can I stop this person from making my invention in the US? No. So in order to stop that person from making my patent in the US, I will have to have a patent in the United States. Okay. So, and it does not happen कि मैंने एक patent file किया और पूरे world में मेरा invention patent हो गया. So I have to obtain a patent. Suppose I want to market my invention in 12 countries in the world. I need to get patents in all these 12 countries. And I have to individually get the patents. एक ही पेटेंट फाइलिंग करके मुझे पेटेंट्स नहीं मिल जाते. Why so? Because every country has a different patent law. What is patentable in country A may not be patentable in country B. तो ऐसे होने की वजह से I will have to go and get patents in all 12 countries. There is a simplified procedure which is called PCT or Patent Cooperation Treaty, under the umbrella of WIPO. Which provides a system to file a single patent application in 140 countries. Now, please try and understand that PCT application is not a global patent. It is just an easier process to file a patent in multiple countries. So when I file a PCT, I will be able to suppose designate 12 countries in the same patent, but that does not ensure that I'll get 12 patents. It can, in all the 12 countries, it can happen that after filing a PCT in 12 countries, I receive patent in 10 countries and the patent gets denied and remaining 2 countries. So PCT again is not a global patent. It is just a simplified mechanism, simplified procedure to be able to file multiple patents. Now, the most important question is, what is patentable and what is not? So if I'm an academic researcher and if I have invented something, I must be able to find out कि मैंने जो invent किया है मेरे lab में, उस पे मैं patent मुझे patent मिल सकता है या नहीं? If I am eligible to get a patent, then I should go for patent first. If I am not, then I straightway should go ahead and file a research paper. So let's see in depth what is patentable and what is not. First and foremost, patents are given on inventions and they are not given on discoveries. So for example, what you see here is a schematic representation of Newton's third law of motion. Action and reaction are equal in magnitude and opposite in direction. So Newton's third law of motion is a discovery. Newton did not make this law in the lab. Okay, it always existed in the nature and he just discovered it accidentally. So he cannot own it. There is something called as sweat of brow doctrine. Sweat of brow यानी क्या? मराठीत ज्याला आपण निधळाचा घाम गाळणं म्हणतो ना. So you have to do effort. You must have taken some efforts and invented something and then only you get it. Discoveries are something which you accidentally find out. You have not made them. They always existed. The nature has made it and you just discover it accidentally. So it is a give and take business. You take efforts, do research, invent something and get a patent in return. Okay. So patents are given not on discoveries, but on inventions. Inventions are those which did not exist before you made it yourself. Rocket works on the principle of Newton's third law of motion. We know that. Okay. But can you get a patent on a rocket? Yes. Because it is an invention. The rocket you have made for the first time, they did not exist in nature. Okay. So rocket is an invention, but the underlying principle, which is Newton's third law of motion, is a discovery. So you can't get a patent on such discoveries or theorems. You get a patent on invention. So basically, patents are given on applied technological things. Okay, it has to be a technology. It has to be a applied science. It cannot be a basic science. So patents are given on invention and not on discoveries. So suppose you are a pharmacognosy professor and you went on a trek and you discovered a new species of some plant, okay, which you have not seen before. Neither your professors have seen it before. So can you file a patent on this? No, because have you made that species? No, it existed in nature. You just have discovered it. So you can't get a patent on discoveries. You have to invent it to be able to get a patent. Now, how do I know whether my patent, whether my invention is patentable? So this is a definition of invention for the purpose of Patent Act. This is an definition of invention from the Indian Patent Act. Indian Patent Act says that invention means a new product or process involving an inventive step and having an industrial application. So it has to be a product or a process. It has to be new. It has to have an inventive step, and it has to have an industrial application. So invention must relate to a process or a product or both. If it is neither a process to make a product nor it is a product, then it is not patentable. It has to be novel. It has to be new. We'll talk about this at length. Okay. It has to involve an inventive step. Another name, these three, novelty, inventive step, and industrial application, are called the patentability criteria. So it has to be new. Inventive step is also called as non-obviousness. So in the US, you call it non-obviousness. In India, you call it inventive step. Another name for industrial application is utility. So in the US, you call it utility. In India, you call it industrial application. Novelty, inventive step, and industrial application are the three criteria of patentability. But before that, it has to be an invention and not a discovery. It must relate to a process or a product or both. And lastly, it should not fall under non-patentable subject matters. We'll see this one by one. It is very important to understand the meanings of these criteria so that whenever you come up with an invention in your lab, you are able to assess it yourself whether it is patentable or not. Just a moment, excuse me. Okay, so the first, we talked about process or product or both. Now let's see one by one the criteria. So when do we say that the invention is novel? Okay, I work as a independent freelance patent consultant, and many times it happens that some individual inventor comes to me and he wants to file a patent. So the first question I ask him is, do you think your invention is novel? Do you think it is new? So his immediate answer would be yes, of course it is new. Then I ask, how do you know that it is a new invention? How do you know that it is novel? He'll think for a while and he'll say, देखा ही नहीं है ऐसे कुछ कहीं कभी. Then I'll ask, देखा ही नहीं है कहीं यानी कहाँ नहीं देखा? आपने कहाँ जाके ढूंढा ये invention? In order to be able to say that my invention is new, you must have checked, you must have searched it before, and then you have come to a conclusion that it is new. So how did you check it? कहाँ देखा नहीं आपने? नासिक में नहीं देखा है, या इंडिया में नहीं देखा है, या दुनिया में नहीं देखा है कहीं भी? And he would not be able to answer it. So when do we say that our invention is novel? Number one, it is not published in written publication. It is not disclosed orally. It is not being sold. It is not offered for sale, nor it is otherwise known to the public anywhere in the world. Okay. Now let us see one by one. What do they mean? Number one, the invention must not have been published in written publication. Okay. So written publication yani kya? It should not be, there should not be any patent on that invention, already published patent, nor there should be any research article. It is not disclosed in any written publication. Written publication can be a patent, it can be a research paper, it can be a newspaper article, it can be a book article, or anything which is written. So if before filing the date of the date of filing your patent, somebody has disclosed the similar invention in a written publication, then the novelty is lost. Okay, now this last sentence, anywhere in the world, is applicable to all these 5. So if it is published in written publication anywhere in the world, the novelty is lost. Okay, then you should realize that the world does not publish only in English or Marathi or Hindi. There are written documents available in several languages. So then if you want to check whether your invention is novel, you should be able to have access to patented documents, non-patented documents, which are in all languages, or you should be able to translate them. And then only you would be able to ascertain the novelty of your work. Okay, so you will have to check in written publications, which can be a patented written publications or non-patented. Then secondly, you should it is not disclosed orally. So your patent is not disclosed orally in any written. Nobody has orally talked about it. Okay, now it is very difficult to prove the oral disclosure. How will you prove that somebody has orally said this before? But nowadays, suppose if you are speaking in a conference, you are presenting a paper in a conference, and there you disclose it, then you often have an abstract of the paper which gets published later, or one can have a video recording of the conference, or suppose you are facing a PhD defense, okay, and you speak about your invention in the PhD defense, which has been video recorded. So if the oral disclosure can be proved, then even the oral disclosure can kill the novelty of the invention. Written publication also means your PhD or master's thesis. So if you feel that you have invented something valuable as a part of your master's or PhD dissertation, then you should first file a patent and then submit the thesis. While you submit the thesis to the university, you get the date stamp on it. So if that date of submission is prior to date of filing your patent, then you yourself have killed the novelty. So अगर आपको लग रहा है कि आपने कुछ तो भी valuable, patentable invent किया है, do not publish a research paper, do not submit your thesis before filing a patent. Do not speak about it in any conference. Do not face your PhD defense. Do not even disclose it to your closest friend before you file a patent. Okay. So thirdly, it is not being sold anywhere in the world. So nothing has been written about it, nor anybody has disclosed it orally, but it is available in the market for sale somewhere in the world. So that also means it is known to people. So sale also kills the novelty. It is offered for sale. Now, offering for sale means it is displayed in an exhibition. Okay, so when do you exhibit it? When you want to sell it in the future. So if it is being offered for sale, then also the novelty is killed. So again, you should not display the model of your invention in any exhibition or modeling competitions or avishkars. So if you intend to file a patent, first file a patent and then display it, then exhibit it in an exhibition. Otherwise, the novelty gets killed. Last but not the least, it is otherwise known to the public anywhere in the world. Okay, so it is neither disclosed in a written publication, not disclosed orally, it is not being sold, it is not offered for sale, but still people know it. What is this? This is called as traditional knowledge. So a classical example is haldi. We know that when my daughter cuts her finger while doing something, the first thing I do is I press a pinch of turmeric on her finger. Why do I do it? How do I know that haldi is a medicine? Because I have seen my mother doing it. My mother has seen her mother doing it. So the knowledge that haldi is an antiseptic is being passed on over generations in India. That is what is called as traditional knowledge. So though we don't buy haldi because it is antiseptic, it is not exhibited, we don't speak about it, nothing has been written about it, but we still know that it is antiseptic. And if this is a traditional knowledge in India, so if you try to file a patent on this traditional knowledge, if you try to file a patent on antiseptic use of haldi, it would not be given because it is not novel anywhere in the world. So if you try to file a patent in India on a herbal formulation which is being practiced by a Adivasi community in Brazil, then also it is their traditional knowledge and we can't file a patent on that. The second and very important criteria is non-obviousness or industrial application. Now, it is very important to understand this criteria for an academic researcher, because non-obviousness or industrial application is a highly subjective criteria, whereas novelty is a very objective criteria. Novelty hoti hai ya nahi hoti hai. Okay, either your invention is novel or it is not novel. There does not, you don't have anything like partial novelty, just like sterility. A product is either sterile or it is not sterile. It cannot be partially sterile. Similarly, novelty is there or it is not there. It is a very objective criteria. Non-obviousness is a very subjective criteria, and therefore it is highly litigated criteria. दुनिया भर में जो सबसे ज्यादा केसेस, कोर्ट केसेस होते हैं पेटेंट्स पे, वो सबसे ज्यादा इस क्राइटेरिया की वजह से होते हैं, बिकॉज इट इज वेरी सब्जेक्टिव, इट इज वेरी ambiguous. So non-obviousness as a patent term means that the invention is not obvious or apparent to a person having ordinary skill in the art relating to the field of invention. Please try and understand what do I mean here. So I'll give you one example. This is an example only. This will not happen actually when it comes to a patent. So all of us understand medicinal chemistry. Suppose आप एक chemist है and you have invented a new molecule in your lab which you feel has some medicinal activity. Okay. Now all of us know that जो एक class होता है medicine का, उनका जो basic structure होता है, the nucleus or the ring structure is same, and we just go on adding substituents. Once that basic ring structure is invented, then as we go on adding different substituents at different positions, we get different medicines, right? And we know that. So suppose you are a medicinal chemist and you have invented a new molecule which is, uska jo basic ring structure hai wo to purana hi hai, but you have added some substituents to that. Okay. Now after you invented it, you were working on it for last 6 years, and then you feel that you should file a patent on that. So when you go for filing a patent on that, and when you do the literature search, you realize that someone has already synthesized the same molecule, same structure, chemical structure जिसका था, वो किसी और ने आपके पहले synthesize किया है और उस पे patent भी लिया है. So obviously your invention is not novel, but because you have spent so many years on doing it, you are bent upon having a patent. Okay, then what you will do? You will do a small change in that chemical structure. आपको पता है, आप मेडिसिनल केमिस्ट हो, तो आपको पता है कि अगर आप एक सब्स्टिट्यूएंट थोड़ा सा चेंज करोगे तो एक्टिविटी में कुछ ज्यादा फर्क नहीं पड़ेगा, बिकॉज यू आर अ मेडिसिनल केमिस्ट, यू नो दैट. Okay, so what you do is, in the original structure there is a methyl substituent at a particular position on that ring. What you do, because of your having basic knowledge in medicinal chemistry, you replace this methyl with an ethyl. Okay, the methyl in the patent, in the original patent, only methyl group is patented. ऐसे होता नहीं है. That's what I said, that this is a hypothetical example. Generally, if I file a patent on that, I will write it as R, and R can be methyl, methyl, propyl, and so on. I'll give you a huge list, and everything I'll secure by filing a patent. But in this situation, ऐसे हुआ नहीं है, और जिसने पहले पेटेंट फाइल किया है उसने सिर्फ मिथाइल सब्स्टिट्यूएंट वहां पे है. So what you do is you simply replace it with ethyl, and rest all the substituents, everything is same. The only difference is methyl. So once you, so when you have changed the chemical structure in order to get the patent, you obviously now will fulfill the criteria of novelty. Novelty criteria कैसे check होता है? They will match with the prior art. Prior art यानी whatever literature is present before the date of filing a patent is called prior art. It can be a research paper or it can be a patent. Okay, तो उसमें क्या है? The ring structure having methyl group. उस जगह पे, उस पोजीशन पे इथाइल ग्रुप वाला किसी ने पेटेंट किया है क्या, या उस पे रिसर्च पेपर किया है क्या? नहीं किया. So is your invention novel? Yes, because the same molecule does not exist before you manufacture it. But the second question the patent examiner would ask is whether your patent is non-obvious. Okay, this is a mistake. It's not industrial application. I meant to write inventive step. Okay, so please read it as non-obviousness or inventive step. These are the synonyms. The second question the examiner would ask whether your invention is non-obvious. So how does he define it? The small change from methyl to ethyl in your invention, is it obvious to whom? The invention shall not be obvious to a person having ordinary skill in the art. Okay, art does not mean painting, sculpture here. Art is any technology. Okay, so which art are we talking about here? When we are talking about synthesis of a medicine, we are talking about medicinal chemistry. Okay, now a person having ordinary skill in the art of making medicine, so that person is a medicinal chemist. So is it obvious for a medicinal chemist, not for a layman? Layman would not know that you can always replace methyl with ethyl and you will get the same therapeutic efficacy. No, a common man will not know that, but a medicinal chemist knows it. Okay, so is it obvious for a medicinal chemist to replace a methyl with ethyl, or is it non-obvious? Obviously, it is very obvious for a medicinal chemist, because all of us know that methyl and ethyl will show similar properties. So though by replacing methyl with ethyl, you have escaped the first criteria of novelty, you will be caught at the second criteria of non-obviousness. And the inventor will say, now while examining the patent for non-obviousness, the patent examiner has to think from a perspective of a medicinal chemist. In this situation, a medicinal chemist, think like a medicinal chemist and decide whether it is very obvious for a medicinal chemist to replace methyl with ethyl or it is non-obvious. So if the answer is obvious, then he will deny a patent. But जैसे मैंने बोला कि novelty is very subjective, non-obviousness is not very objective, non-obviousness is very subjective, क्योंकि हर आदमी के IQ में difference होता है. So एक अगर बुद्धू मेडिसिनल केमिस्ट है, उसके लिए कोई चीज नॉन ऑब्वियस होगी, और वो अगर इंटेलिजेंट है तो उसके लिए ऑब्वियस होगी. सो हु इज द फ्लोसीता, वहां से ही इसके डिस्प्यूट्स चालू हो जाते हैं, एंड इसलिए इट बिकम्स वेरी डिफिकल्ट फॉर द कोर्ट्स टू प्रूव. तो अगर एग्जामिनर बोलता है कि ये ऑब्वियस है, and if you appeal it to the district court judge will say that it is non-obvious. Then the Supreme Court says it is obvious, and it goes on. So non-obviousness is the most litigated criteria of patentability. The Indian Patent Act, which calls it inventive step, defines inventive step as it involves either a technical advance as compared to the existing knowledge. No, जो existing knowledge है That is called as prior art. So your invention should have a technical advanced over the existing knowledge, or it should have an economic significance over the existing knowledge. So agar you have replaced methyl with ethyl, you have to prove that by replacing methyl with ethyl, there is a tremendous enhancement in therapeutic efficacy, which we can call as technical advance. Or I should be able to prove that making a ethyl substituent is very, very cheap compared to methyl. So either it has a technical advance or it has a economic significance, or both. If you can prove that, then it becomes non-obvious. Otherwise, it remains obvious and it does not get a patent. The third criteria of patentability is industrial application or utility. So when we are talking about pharmaceutical patents, we always have an industrial application. Okay, medicines can always be made on an industrial scale. So this is not a very difficult criteria to understand. But again, if you have discovered something like Newton's third law of motion does not have industrial application. Newton's third law cannot be made in the industry. You can make a rocket, which is a product based on Newton's third law of motion. But to be able to get a patent, your idea you should be able to make it in the industry. And as I said, you get patents on technology, you get it on applied science, and you don't get it on basic science. So this is a third criteria of patentability, which is called industrial application. Then C. Okay, so you have made something in your lab, and the questions you want to ascertain the patentability of your invention. So the questions you ask is it a process or a product? The answer is yes, it is a product. Then is it novel? Yes. Does it have inventive step? Yes. Does it have industrial application? Yes. So you fulfill all these criteria. Will you get a patent then? Generally, yes, but you may not still get a patent if your patent falls under the non-patentable subject matter. Now, ye kya hai? So, though your invention satisfies all the criteria of patentability, you may still not be able to get a patent. For example, life forms, living organisms are generally considered non-patentable all over the world. Softwares as such are considered non-patentable, but if the software is claimed as a part of hardware, then you get a patent. For example, agar aapne Microsoft Windows is a software per se, software as such. So can you get a patent on Microsoft Windows? No, you will get a copyright. But suppose आपने एक विस्कोमीटर लिया है लैब में, और विस्कोमीटर के साथ एक रेओकैल्क नाम का सॉफ्टवेयर भी आता है. So if you claim रेओकैल्क as a part of viscometer, then software as a part of hardware may get a patent. So आपने patent file किया है वो viscometer पे, उस hardware पे, and software is one part of that hardware, you may get a patent. But software as such are not patentable. Business methods are not patentable. For example, methods of doing business, like you have heard about the Amway business or Tupperware business, multi-level marketing. So it is a method of doing business. You will not get a patent on this. I would like to throw some more light on this, because this is more relevant to pharma, that how the jurisprudence of patentability of living organisms has evolved. So when patent laws, these are some examples of living organisms, and this is how the jurisprudence has evolved. So initially, all the living organisms were considered non-patentable. Why? All the living things, either organism or plants, were considered non-patentable, because it was considered that living organisms are made by nature. Man cannot make a organism, right? Because the patent laws were made in 18th century, and at that time the lawmakers did not know that how much the biotechnology is going to evolve in future. So this is a classical case, which is say Diamond versus Chakravarti. Chakravarti, Anand Mohan Chakravarti, was an Indian staying in the United States of America. He was a genetic engineer working for General Electric. He developed a bacterium. This is Chakravarti's bacteria. He developed this bacteria which was capable of breaking crude oil, this bacteria could actually eat crude oil, and therefore Chakravarti said this bacteria could be very useful in treating oil spills from the big ships. You know कि जब वो जहाज जाते हैं तो उनमें से oil spill होता है. So if you culture these bacteria on these oil spills, they would be able to eat this up. This is called Chakravarti's bacteria. Now, this bacteria was made by Chakravarti in the lab. This did not exist before. He made it by genetic modification. So this was a genetically modified microorganism, and it did not exist in nature. It was not a discovery. It was an invention. Chakravarti made it in the lab. So GE Healthcare filed a patent in the US with Chakravarti as an inventor on this bacteria. So obviously at that time the jurisprudence was living organisms are not patentable. Diamond was a examiner in the US patent office, and he denied the patent on this. Chakravarti claimed that it was rejected by the examiner because at that time it was generally understood that living things are not patentable. Chakravarti, what did he claim in the patent? He claimed the process of producing the bacteria, and he claimed the bacteria itself. Okay. So then Chakravarti appealed against this examiner Diamond, and hence the name of the case is Diamond versus Chakravarti. So at that time, the United States Code said, section 101 is about novelty. It said that whoever invents or discovers any new and useful process, machine, manufacture or composition of matter, or any new and useful improvement thereof may obtain a patent. So कोई भी अगर कोई भी नई चीज, new and useful process, machine, manufacture, composition of matter, they make it, then he can get a patent. So Chakravarti said that I have manufactured this organism. Neither it is a law of nature, it is not a physical phenomenon, nor it is an abstract idea, because these three things were prohibited. So he said, my invention fits in here because I have manufactured a microorganism. I have manufactured the bacteria in the lab. So I must get a patent. So though Chakravarti's microorganism was a living microorganism, for the first time, a patent was granted on a living thing. Okay. So then how the jurisprudence changed? Before Chakravarti, it was understood कि कोई भी चीज अगर living है तो उस पे patent नहीं मिलेगा. But after that, the court said that even if it is a living thing, if it is man-made, then it can get a patent. So microorganisms after this became patentable. Then came the second case of Harvard oncomouse. This is an oncomouse which was a transgenic animal. It was made by the scientists in the University of Harvard in 1980s. They produced this genetically modified mouse, and it was highly susceptible to cancer. So इस mouse में cancer induce करना बड़ा आसान था, and therefore it was called a oncomouse. So they genetically modified it so that you would be able to easily induce cancer, and then this can be used in the preclinical studies of oncomedicines. This was a transgenic animal. A transgenic animal is one when DNA from the other species has been artificially introduced into their genome. So after Chakravarti, microorganisms had become patentable, but this was a larger animal. This was a mammal. So again, when Harvard scientists, you mute? Yeah. This patent, when Harvard scientists filed a patent on this this mammal, the patent was again denied, saying, can a large animal like mouse be patented? And then again, they proved that this was a transgenic animal. It did not exist before. They have manufactured it, and then the patent was granted in the US. Diamond versus Chakravarti case became the precedent, and they said that they claimed that it is similar to Diamond versus Chakravarti, but he manufactured a bacteria, we have manufactured a mouse. So this patent was granted in the US, but it was again granted in the European Patent Office, but the Canadian Patent Office rejected it, saying higher life forms are not patentable because they cannot be considered as manufacture or composition of matter. So I said it before that patent rights are territorial, and what is patentable in country A may not be patentable in country B. This is a classical example of that. So patent was granted in the US, granted in the EU, but denied in Canada. Then came the next case of biological matter. This case is called Association of Molecular Pathology versus Myriad Genetics. So you might have heard about this BRCA gene. So BRCA gene, these scientists, the scientists from Myriad Genetics, they claimed that if you see a mutation in BRCA gene, then the person is likely to get breast cancer. Okay, so अगर आपको दिखता है कि इस BRCA gene में mutations हो रहे हैं, so that female is likely to end up into breast cancer. So this can be used as a diagnostic test. Okay, so what they did was, the Myriad Genetics scientists, they said that they announced that they had located a gene on the chromosome 17 that provides the first evidence of the connection between certain genetic variations and breast cancer. They sequenced, the scientists at the Myriad Genetics, then what did they do? They do the DNA sequencing of BRCA1 and BRCA2 genomes, which were responsible for breast cancer. And they developed diagnostic test. They obtained patents covering the sequence, covering the, apart from many other things, they also got a patent on this DNA sequence. And they claimed exclusivity over these tests then, the diagnostic tests. And this patent was challenged by Association of Molecular Pathology. What did they claim? They said that this patent should not be granted. Because what has Myriad done? They have just, have they made a DNA? No, they have just sequenced, they have just isolated a DNA which is responsible for breast cancer. So Myriad has isolated a DNA and they have not created it. Is this a discovery or invention? This is a discovery because the DNA is made by nature. DNA is not made by Myriad scientists. So can a human gene be patented then? And if the human genes become patentable, then what are going to be the moral implications of this? So the judgment said that separating the gene from its surrounding genetic material is not an act of invention. The court, while saying, they said that अगर आपको किसी पेड़ पे पेटेंट नहीं मिलता है तो आपने क्या किया, उसकी एक डाली तोड़ के लाई. Because you cannot get a patent on a tree, you have excised a twig from the tree and you are trying to claim a patent on that. So this would not be given. So DNA isolation or DNA sequencing is supposed to be a discovery and not an invention, and patents are not given on that. So this was all about the jurisprudence of patentability of life forms. So initially they were considered non-patentable, then they gave a patent on a genetically modified bacterium, then they gave a patent on a genetically manufactured mouse, which is a mammal, and then they denied a patent on DNA isolation. Another issue in patents, patentability of living forms is a moral implication. So this is a scientist called Oliver Brusley. He was a European scientist and he filed a patent on obtaining nerve cells from human embryonic stem cells. So he tried to, he used a 2 weeks old human embryo. He isolated stem cells from that embryo and he tried to obtain nerve cells from these stem cells and filed a patent. He got a patent in the US, but German laws prohibit the use of human cells for industrial use. So the European Court of Justice said that you have killed a 2-week human embryo to be able to invent this. So you have killed a life and it is very immoral. He claimed that it was just a 2 weeks old embryo and not a fully grown human being. The European Court of Justice said that a 2 weeks old embryo has the equal right to life like even a 1-day-old embryo has the same right to life as that of a fully grown human being. And therefore this patent was denied, but it was not denied on the novelty or non-obviousness or industrial application ground, but it was denied on morality ground. And he said that if we allow patents on this, then people will start killing embryos and start using them for research purpose, which is not allowed because the embryo has a right to life. Okay, so that's all about the patentability of life forms. We'll quickly go through. Do we have some time or we need some time for question and answers? I can stop here.
Ma'am, we can go for discussion also, no issues.
Okay, so I think I'll stop here and we'll take questions on because this was about the patentability of plant-based things, but we can skip that. Okay, I'll quickly go through this part. I said that naturally occurring substances are generally not patentable because they generally are a part of traditional knowledge. तो फिर आपको ये क्वेश्चन हमेशा आता है कि does that mean that one cannot patent a natural product? No, it doesn't mean that. Then what type of natural products are patentable? So formulation of a new composition or an improved formulation can be patented. So though the active ingredient is known, traditionally known, if you have developed a new formulation, for example, a herbal anti-allergic composition, which is a synergistic mixture of extracts of fruits of Terminalia chebula, albizia, and Terminalia bellerica, and the process of preparation of such composition. So if the formulation is novel, you can get a patent on the formulation. If it is, if you are using an old herbal constituent for a new use, the new use is not known. Okay, so this is a how do we pronounce? Forskolin can be used for weight loss, and it was discovered for the first time by Sabinsa Corporation. This is not a traditional use of forskolin. This is a naturally occurring ingredient. Which was not known for this use. So if you have invented a new use of a known compound, plant-based medicine, then that can also be patentable. Patentable natural products like novel glycosides extracted and isolated. So if you have modified or synthesized, you have developed a synthetic route for making a natural product, then that can be patentable. But whenever you want to file in India on a naturally occurring medicine, then you first have to get a permission from the National Biodiversity Authority in this form. So you have to get their permission and you have to attach this permission to your patent document. Then only you would be able to file a patent. And last but not the least, I told you that life forms, softwares and business method are considered non-patentable all across the globe. Okay, but in India, what are the things which are considered non-patentable? Briefly, an idea that is frivolous or which claims anything contrary to a naturally established law. So for example, you try to claim a patent on an instrument or a machine in which output is more than input, so which is contrary to the natural law, nature's law. We know that output cannot be more than input. So if you are trying to claim something which is contrary to it, you will not get a patent. If you have invented something whose intended use is contrary to law or injurious to public health, for example, you try to make a diagnostic test, which is a very easy method to diagnose the sex, to determine the sex of a fetus. Okay, so it can be morally wrong, because we know that in India is banned, so it will not get a patent in India. If you have merely mixed substances, there is just additive effect and no synergistic effect, you will not get a patent. Mere discovery of a new form of a known substance. So you just have changed a new form. For example, you have invented a new polymorph or a new isomer of a known drug. There is no enhancement in therapeutic efficacy. Just a new salt which has better compressibility or better stability, but no enhancement of known efficacy. This new form is not patentable in India. Methods of medicinal surgical methods or operation surgery methods are not patentable. Anything which is a process related to agriculture and horticulture is not patentable. Mathematical methods or method of solving a puzzle or playing a game or a business method or a mathematical algorithm or a computer program is not patentable in India. Mere scheme or rule or method of performing a mental act or method of playing game. Anything which is related to atomic energy, so inventions related to uranium, beryllium, thorium, radium, graphite, they are banned from patenting in India. Topography of integrated circuits cannot be patented, and traditional knowledge cannot be patented. Okay, so these were some another thing, important thing is, if you happen to file if you have published a paper, and after publishing a paper, obviously you have lost the novelty. If you feel that I should have filed the patent, in India, you get a grace period of 1 year. So if you file a patent within 1 year from publishing a paper, you are allowed to file a patent, but that has to be done within 1 year. If you do it after that, then the novelty is lost and you don't get a patent. Okay, so I feel we should stop here because I have one more session coming up after this. So I'll be happy to take your questions. I hope this has given you an idea about what is patentable and what is not.
मैम मेरा एक क्वेश्चन था कि ट्रेडिशनल नॉलेज में कोई मेंशन नहीं है, वो कोई नेचुरल प्लांट से ही ऑब्टेन्ड आपका मेडिसिन है, उसकी फॉर्मुलेशन यदि हम डेवलप करते हैं और वो फॉर्मुलेशन इफेक्टिव है तो क्या वो पेटेंटेबल है?
That's what I said, that if you have developed a new formulation which is not known before, it is patentable. Okay. But you get a patent on the new formulation only, not on the use.
Okay. Okay, ma'am.
Anybody else having any queries or some discussions you want to do with madam among the participants? Okay, there is one question in chat box.
Yeah. Can nanomaterial synthesis from novel source could be considered as novel? Let me tell you one thing, that we are all belonging to science, and for example, we are used to having unequivocal answers to every question. So hamare liye 2 + 2 4 hi hota hai. But in case of law, it depends is always the answer. So when you ask me a question that is this patentable, I would not be able to answer that हाँ इस पे patent मिलेगा या हाँ no इस पे patent नहीं मिलेगा. It depends. So if does it fill in the criteria of novelty? So even if it is a novel source, is it very obvious source? All these criteria will apply, and then the answer would be yes or no. Many times it depends on your skills to draft a patent, and based on which you either get a patent or you don't get a patent.
Okay, I think the queries are satisfied at least for the first session. Dear participants, I am delighted to announce that Dr. Maria Saifi, Associate Professor, YB Chavan College of Pharmacy, Aurangabad, is chairing the sessions, both the sessions of Dr. Bede Madam. And I think, Madam, we will take a break of 15 minutes if you wish.
Yes. I need a
Okay. So 10 to 15 minutes will just break.
Yes.
You can have a power nap also, but don't leave, be online. So we will meet after 10 minutes. Thank you, Madam. Thank you.
cannot be more than in
Look, man, I just want my sales back.
Participants, we are waiting for Mrudula madam to join back. So bear with us. We will start the session as soon as she joins. Thank you. So dear participants, again, welcome back to the last session of this day, the first day of STTP. I hope you all had a very good power nap. So we are back again. Madam is going to give a talk on the access to medicine and COVID. Are patents a barrier? So it is with the going times, she is having a discussion with us. So I invite Madam again to please start your session. Over to you, Madam.
Thank you. So in the first session, we discussed the general things about patents, that what is patentable and what is not, and etc. In this session, I would like to throw some light about how patents affect access to medicine. And I feel to all of us who are being professors in pharmaceutical sciences, we should know this. And we should make it a point to pass on this knowledge to our students when we teach intellectual property rights, because this is a very important perspective of patents, human rights perspective of patents, which I feel we all should know, and this is a sensitive issue, so we should know about it. So in this session, we'll discuss how patents affect access to medicine in general, and what is the patenting scenario of there is a mistake, apologies for that, and how patents are going to affect that, how they have affected the COVID-19 medicines access, and what has been the Indian approach, and what are the solutions India could have to get rid of these barriers in form of patents. So this in general is the process of drug development, which we all are aware of. Okay, so I'll go through it quickly. That whenever a pharma company develops a new drug, this is a process through which the drug goes. The company chooses the target disease, it identifies the lacuna in the conventional therapy. Then it synthesizes several new chemical entities, does preclinical studies, file patents on the studies which show some promise, does clinical studies, files, gets marketing approval, and finally, after getting marketing approval, the post-marketing phase 4 studies go on. So this is a conventional way by which a new drug or an innovator drug is brought into the market. Now, the drug development process, we know that when we synthesize around 10,000 to 30,000 molecules, one or two molecules are fortunate out of 10,000 to 30,000 to be born as drug. So it's a very tough job. So if we start with 10 to 30,000 molecules, around 10 to 20 enter the preclinical studies, 5 to 10 enter phase 1, 2 to 5 enter phase 2, around 2 enter the phase 3, and so hardly one becomes a drug. Many times none becomes a drug, and the entire process gets wasted. Okay, the entire effort. Now this process takes the pipeline of the research pipeline of drug development is very, very prolonged. It takes around 10 to 12 years to develop a new molecule, and the cost of bringing one new molecule, the average cost of bringing one new molecule to the market was supposed to be around 2.6 billion US dollars. So 1 billion USD are 100 crores USD. Okay, so 1 billion is 100 crores USD. So 260 USD multiplied by 72, 73, whatever is the dollar rate, that is in rupees. So that is the cost of bringing one new molecule, one innovator molecule into the market. And that is the reason when the drugs come for the first time in the market, they are so expensive. Okay. Now, why are the drugs expensive? Because the pharma companies say that was a huge cost of R&D, and that this cost of research includes the API research, that is a medicinal chemistry research, then the formulation research and the clinical research. And clinical research is supposed to be the most expensive one. Then you have to do the patenting. So the patenting is an expensive affair. Obtaining the regulatory approvals is another expensive affair, and the cost of marketing. So the cost of new drug when it comes into the market is high because the cost of R&D is very high. There is a huge cost incurred on patenting, obtaining regulatory approvals, and marketing. Now, out of this 2.6 billion USD, how much amount is spent on R&D, how much on patenting, and how much on marketing? This the pharma companies never disclose. So you will never come to know that what fraction of this 2.6 billion USD was actually spent on R&D and how much was spent on marketing. That is not known. Now, because most of the new drugs are born in the United States of America, I am talking right now only about USA. Okay, so because the first new, because in general, say 7 out of 10 new drugs are first born, remaining are born mostly in the Europe. So because most new drugs are born in the US, what exactly is the role of patents during the process of drug development, or where do they come? So when a new chemical entity shows promising results in animals during the preclinical studies, what you're showing below in this green window are the regulatory pipelines. And what you are seeing above in the blue window are the processes or happening as far as patenting is concerned. So as soon as a NCE shows result in preclinical studies, what does the company do? What does the innovator company do? It files a patent. Now you do not know how many of these molecules are actually going to enter the phase 3 studies. You are not aware. So if 4 or 5 molecules show promise in preclinical studies, the company has to file patents on all of them because it does not know which one is going to succeed. Okay, so it will file patents on all of them. At the same time, it will also file a INDA, that is investigational new drug application. So this is happening at the US FDA in America, and this is happening at the USPTO, that is United States Patent and Trademark Office. Okay, so you file an investigational new drug application to be able to start the clinical studies, that is studies in human beings. You do your phase 1, 2, and 3 trials, which will take around 7 to 8 years to finish these trials. Now, by the time you have finished phase 3 trials, you also are likely to have obtained the patents. Okay, and when you file a new drug application, that is marketing approval at the US FDA. Now, this I am speaking only about the United States of America. This does not apply to any other country. Okay, so when I finish phase 1, 2, and 3 trials and I submit the marketing, the new drug application at the US FDA, I am expected, please listen to this very carefully because this is a unique process which happens only in the US. Okay, so I am expected to enlist all the patents which are covering my drug with the US FDA. In all other jurisdictions, including India, DCGI in India, the regulatory authorities, CDSCO and DCGI, they are not bothered about your patents. Okay, you just have to show them the safety and efficacy data of the phase 1, 2, and 3 studies, and they will grant you a marketing approval. The regulatory authorities in other countries are not bothered about whether you have got a patent, whether you are infringing someone else's patent or not, because they say that is not our job. But in the US, the time of getting marketing approval, the US FDA has to go through all the patents obtained by you, and the pharma company will not have a single patent. It will have several patents. Like the first patent would be on the basic molecule API. Then there would be patents on the crystal forms. There would be patents on the method of use, treatment and dose, and there would be patents on formulations. So the innovator company has to enlist all the patents it has obtained relating to that drug in the with the US FDA, and these patents are listed in the Orange Book. We'll see how the Orange Book. After the FDA goes through your patents, it gets a market. It gives you a marketing approval. It gives the pharma company the marketing approval for the marketing of its innovator drug. So this is how the patents come into the picture here. Okay. Now, because in the United States of America, the marketing approvals are linked to patents, it is called patent linkages. Okay. Now, the practice of patent linkages does not exist anywhere outside the US, but it is very important for us to understand this because most of the new drugs are born in the US. So this is an overlap of IP, that is patents and regulatory. So this overlaps happens at the time of getting marketing approvals, where you have to enlist all your patents with the FDA in the Orange Book. And this linking of marketing approvals to patents is called patent linkages in the US. Now, when the patent expires, we know that life of a patent is for 20 years. So after the patent expires, the generic drugs come into the market. Now, generic company files the ANDA, that is abbreviated new drug application, with the regulatory authority. Now, the generic company does not have to prove the safety and efficacy because it is only a duplicate. It is a Me Too drug. It had just copied the innovator drug. It only has to prove the bioequivalence. So the marketing approval only consists of the bioequivalence data. So it is short, and therefore it is called abbreviated new drug application. And once the NDA gets approved, you have proved that your generic formulation is bioequivalent with the innovator formulation, you get the marketing approval. So generally, generics do not file any patent unless they have developed a new formulation. But the basic molecule patent is always on the name of the innovator, and the generics cannot market their drugs unless and until the basic molecule patent has expired. Okay, now we know that there are the two important features of patents are territoriality. We discussed about this, that what is patentable in country A may not be patentable in country B, and the patents have a limited protection. They expire after it expires. It comes in public domain and anybody can use it. So similarly, when a patent of the drug expires, the generic can use that know-how, that information in the patent. It can replicate and it can market its generic drug. Okay. So what happens when I say that what is patentable in the United States of America may not be patentable in India? Why it is so? Because every country has a different patent law. We saw that. Okay. So but why do all the countries have different patent law? Why can't we have a harmonized patent law like we have ICH guidelines, which are harmonized guidelines for stability studies? So why can't we have a harmonized patent law? That is because whenever you grant a patent on something, what you grant is the monopoly. Okay. Now, when the innovator company has a monopoly on the drug, then there is no competition in the market. Nobody else can sell the drug without except the innovator company. And so when there is no competition in the market, the costs of the patented good are going to skyrocket. They will be very heavy. The drugs are very expensive because there is no competition in the market. So the innovator company actually sells the drug at muh bola daam, jitne chahiye utne price mein they start selling their drug. Okay. Now, when they do this, So if you make your patent laws, the patent laws of a country are such easily give away patents, then what will happen? They will create more monopolies. More is the monopoly, easier is the way by which you can obtain a patent. And so what happens? More and more things, if they are get a patent, they become expensive and they get out of the reach of a common man. So whenever you grant a patent on something, the thing becomes unaffordable to a common man. So in a country like United States, where per capita income is very high, the patent laws are such that you can easily get patent. The United States courts themselves have said once that anything under the sun made by man is patentable in the United States of America. Now, in a country like India, if we are talking about a life-saving commodity like a drug, can we afford to make it so expensive to every Indian citizen? Because we know that the per capita income is very low in India. Moreover, in India, the out-of-pocket expenditure is very high because very small percentage of population is covered by medical insurance, right? So most of the population has to spend on healthcare from their own pockets. So in this situation, if you give away very easily medicines, then the patents become beyond the reach of common man and they become expensive. So moral of the story is, if your country's patent policy is favors the innovators, then the things become expensive to common man. So if then public at large is at loss. And if it favors, so in a country like India, the patent laws are very strict. We don't give patents very easily, especially on medicines, our laws are very strict. So the public at large is at favor, but the innovator is at loss. Okay. So a country's patent policy will either favor the inventor or it will favor common public. It cannot favor the balance of the patent law. It will either favor the economic gain to the inventor or it will favor the welfare. Now, the treaty, the international treaty which was governing patents before 1995 was Paris Convention. Okay, so Paris Convention is administered by World Intellectual Property Organization. And the main thing, so the Paris Convention said that patent किस पे देना चाहिए, किस पे नहीं देना चाहिए, कितने year का life होना चाहिए, and all. It was mentioned in the Paris Convention. But the biggest flaw in the Paris Convention was it did not have any enforcement mechanism. So if a country does not abide by the guidelines given in the Paris Convention, what will happen? Nothing will happen. So किसी भी law में एक enforcement mechanism होना चाहिए. So if it is a, say example in India, you have a criminal procedure code. So if you murder someone, this would be the charge on you, and you will go to jail for so and so years, or you will be fined. So that is an enforcement mechanism. Okay, but if it in absence of the enforcement mechanism, nobody will fear the law. Okay, so because there was no enforcement mechanism for the Paris Convention, countries had their own laws. There was no guideline on which the law should have been based, and the patents had different periods of protection. Some countries did not give patents on medicines, some gave. India did not give patents on products, and it gave patents only on processes and so on. So it was a chaos, okay, and every country used to do whatever they felt correct. What was the scenario in India before 1995? We were using the old Patents and Designs Act, which was formed by British. That is a Patents and Designs Act of 1911. And India was giving away both product and process patents for drugs. Sorry, this is, I meant to write before 1970, not 1995. So before 1970, India was giving away product and process patents on drugs, because of which, and the pharma industry in India hardly existed before 1970. Okay, let me just Patents, India was giving away product and process patents on drugs before 1970, but the pharma industry was hardly there. So 85% of the drugs were being imported. Prices were among the highest in the world. Drugs were extremely expensive in India because pharma industry almost did not exist. And the mortality rate was very, very high. One of the reasons why people used to die because of trivial health problems, one of the reasons was the drugs were not available. And why they were not available? Because they were being imported, and so they were very, very expensive. Now, before 1990, again, the same mistake. So before 1970, after 1970, what we did was, in order to improve the access to medicine, we changed our law and we had Patent Act of 1970. So after India enacted this new patent law of 1970, we decided that we will not give patents, product patents on pharmaceuticals. We will give only process patents. So suppose paracetamol is a drug, so paracetamol will not have a patent in India. Only the process to make paracetamol would be patented. So because of that, what happened? There was people were not prohibited from making paracetamol. They were prohibited from using the process which was patented. So what did we know that we Indian people are very good in jugaad innovation. Okay. So what we did was we reverse engineered all the patented drugs. We found out new, jisko aap retrosynthesis bolte ho. So we developed new non-infringing processes of making those drugs. And so, and also for these processes, also they were, the life of these process patents on drugs was only 7 years. Okay. So because the term of process patent was only 7 years and there were no patents on products, Indian pharma industry started reverse engineering all the patent products, and a country which was a largest importer of the drugs before 1970 slowly became the largest exporter of the drug. The pharma industry became self-sufficient by reverse engineering. So during this COVID period, we have heard this several times that India is the pharmacy of the world. Why the pharma industry in India has grown so much? One of the reasons was the Patent Act of 1970. So during the period of 1970 to 1995, Indian pharma industry bloomed like anything and it became the largest exporter of APIs and largest exporter of generic formulations. So India became the largest exporter of generics. Now, then came the TRIPS agreement of 1995 because the big pharma, that is the multinational pharma companies like Pfizer and Eli Lilly and AstraZeneca, GSK, which are all based out of the US and Europe, they realized कि अरे हम इतना research करके नए molecules develop कर रहे, नए innovator molecules market में ला रहे. And what India is doing, the Indian pharma is reverse engineering because patent laws are territorial. So it is completely legal in India to reverse engineer because there are no product patents. So they were at loss. They were spending billions of dollars on R&D of the new molecules. And as soon as their new drug came into the market, the Indian pharma developed a non-infringing route to synthesize that molecule, which was legal, completely legal in India. So in order to stop this practice, they thought of devising a new agreement, which is called TRIPS agreement or agreement on trade related aspects of intellectual property. Now, this agreement was proposed and it was passed in 1995. And what happened was, just before this, WTO, the World Trade Organization, was formed. So before this, before 1995, every treaty or international agreement on intellectual property was administered by WIPO. and none of them had any enforcement mechanisms. But in 1995, all the countries, especially US, Europe, Japan, Australia, they proposed this TRIPS agreement, and they said that here onwards, this agreement would be administered not by WIPO, but it would be administered by World Trade Organization. And those countries who want to become members of WTO must become signatory to TRIPS. It was mandatory, and countries were very keen on becoming members of WTO because it was needed for trade. And if you want to become a member of WTO, you must become signatory to TRIPS. So India and Brazil were the two countries who opposed the TRIPS agreement, but it was finally it was a coercion, and India had to become signatory to TRIPS. Now, why was India resisting TRIPS agreement? Because TRIPS agreement provided criteria for minimum protection. Now, I told you that before that, every country had a different period of patent term. Like in India, it was only 7 years for pharma patents. In some countries, it was say 15 years. But TRIPS agreement said that the minimum life of a patent has to be 20 years. Country can go above 20 years. They can provide protection which is more than what is mentioned in TRIPS agreement, but it cannot be less than the minimum protection. So the term of patent was increased to 20 years. It also required introduction to both process and product patents on all fields of technology. So what we were doing in India, we were not giving product patents on pharma. TRIPS agreement said nothing doing. You have to give both process and product patents, and you cannot discriminate based on the field of technology. We were discriminating. We were saying that we'll not give product patents on pharma. We'll give product patents on others. TRIPS said that this would not do. You will have to give process and product patents on both the fields of technology. Now, for countries like India, for developing countries like India, TRIPS agreement provided a period of 10 years, a transition period of 10 years, so that they said that though you are becoming signatory to TRIPS in 1995, you have to become completely compliant with TRIPS by 2005. So you have got 10 years, because in order to become compliant with TRIPS, you will have to change your laws. You will have to amend your laws. So we got a period of 10 years up to 2005. And in order to become compliant with TRIPS, India amended its patent law in 1999, 2002 and 2005. In 2002, we increased the term of patent to 20 years. And in 2005, we started giving away patents, product patents on pharmaceuticals. Till 2005, there were only process patents on pharmaceuticals in India. Now, the TRIPS agreement specifies certain minimum standards of protection. As I said, you can go beyond that. A country can go beyond that, but it cannot be below the minimum standards. So the countries which provide more protection than that is mentioned in the TRIPS agreement for the TRIPS plus provision. So obviously, countries like countries like US and Europe and Japan and Australia, they want to give more protection to their inventors, to their industry. So they use TRIPS plus provisions. The first I mentioned before, that is patent linkages. Another is patent term extension or supplementary protection certificate. So patent term extension is a term used in the US. So they can increase the term of patents beyond 20 years. The same is called SPC or supplementary protection certificate in the EU. And one more one more protection is called data exclusivity. So we'll see one by one, because see, remember, the generic company cannot come into the market unless and until the patent expires. Okay. And it is very essential for a common poor man or a man having average per capita income. It is very important that they have access to generic medicines. Unless and until the generic entry happens in the market, the prices of the medicines do not go down. So it is very essential that as soon as the patent expires, within 20 years, the generics come into the market. Now, what happens that because of these TRIPS plus provisions, the life of a patent is increased beyond 20 years. We'll see how. And so it delays the entry of the generic medicines in the market. And in turn, what happens is the medicines become beyond the reach of common man because they remain expensive. The generic entry does not happen after 20 years. It is further delayed. Okay. So patent linkage, I told you that when the drug approval process with patent, it is called patent linkages. So patent linkage refers to the communication between the national regulatory authorities and the patent office. So it is US FDA and USPTO in the US to prevent marketing approval of generic drugs until after the expiration of patents covering the drug product or approved use. So in India, if a generic, there is a innovator product in the market, suppose, and the generic company wants to launch its product, it files a clinical study data with the CDSCO or DCGI. The DCGI feels fit that the drug is bioequivalent with the innovator, it will grant the approval. It will not check whether the innovator patent has expired on the drug or not. Okay. So if the generic approval has been granted, despite the patent being alive, it is not the responsibility of DCGI. Then suppose अगर आपका generic approval हो गया और आपने market में लाया आपका drug and the innovator patent is still alive, then what the innovator company does, it will have to file a suit on the generic company. The fight will go on in the courts. And if the innovator company wins, then the generic company will have to take back the product from the market. Okay. But the regulatory authority does not look into the patenting of the medicines. Whereas in the US, the regulatory authority will not grant a marketing approval, will not grant a ANDA to the generic company unless and until it ensures that the patent of the innovator is expired, and then only the generic company can come into the market. So new drug application must include the patent information, and the FDA considers the existence of patents as part of the approval process. So the generic marketing approval is linked to the expiration of the pioneer drug patent or the innovator patent, and that is what is called as patent linkages. So this is available in the Orange Book. Let us have a look at how does this Orange Book is nothing but a record of all this information maintained by the US FDA. So is this visible?
Yes, madam, it is visible.
So this is the Orange Book. The other name for Orange Book is the approved drug products with therapeutic equivalence evaluations. Okay. So this is, you can easily find it on the US FDA website. So suppose one drug called pitavastatin calcium. Okay. So I want to check the regulatory approval status of this drug. So what I do, I type in this name and search it. So what are the drugs approved for pitavastatin calcium? You can see. So can you see here RLD? RLD means reference listed drug. Okay.
Ma'am, your page is not visible.
You'll have to share.
Nobody is able to see it.
The Orange Book page
is not
visible.
The screen is not actually the PowerPoint presentation is visible to us, but the Orange Book, your desktop is not appearing. That window is not appearing.
Stop the presentation and share this window.
Might be.
Now, can you see?
It's working, madam. It has started. Yes, we can see it.
Yeah. So I just searched pitavastatin calcium on the Orange Book. So can you see here? There are several products which are approved. Okay, you can see several Indian companies like you will see Mylan, you will see Aurobindo. So these are the generic products. You see RLD before this product, which is called Livalo. So RLD means reference listed drug, which means this is an innovator drug. by, and who is the innovator company? Kowa Company Limited. So this product was first invented by Kowa, and after the patent has expired, these generic products are available, but this is a reference listed product. Reference listed means when these company file their ANDAs, they have to compare, they have to do their bioequivalence with these reference listed products. You can see three are reference listed products because these are three strengths: 1 milligram, 2 milligram, 4 milligram. So the application number is this. So when I click on this, yes. So here you see, can you see this line, patent and exclusivity information? So when I click on this, you will see there are 5 patents on this drug. So this is listed on the US FDA website. So before granting, so this is the first patent. This is a basic molecule patent which is expiring this year, this month. So 25th June, this patent expires. Okay, so these are the patents, and one is a crystal polymorph patent which expires in 2024. And this is a method of use patent which expires in 2024 again. Okay, on 2nd February and on 19th August. Okay, what you see here, PED, is the pediatric exclusivity. We'll speak about this later. So what I wanted to show you is this is how the patent linkages happens. So the US FDA in the Orange Book has to mention all the patent related data. There are 5 patents or 5 exclusivities, and they are expiring on so and so date. So the generics will have to refer to these patents and do their applications accordingly. Okay, I'll stop this and we'll come back to the presentation. Okay, so this is what is called as patent linkages. Then another TRIPS plus provision which exists in the US as well as in the EU. In EU, as I said, it is called special SPCs, and which is supplemental protection certificates. In the US, it is called patent term extensions. So in US, the patent does not expire, may not expire after 20 years. The owner of the patent, this exists only in case of pharma and food. So if we know that though the life of a patent is 20 years, 7 to 8 years are lost in getting the drug regulatory approval. So the effective term which the innovator gets is only of 10 to 12 years, excuse me. So the innovators say, the innovator pharma companies say that हमको तो सिर्फ 10 या 12 साल ही मिलते हैं और उतना ही मोनोपोली मिलता है. 20 साल तो मिलना चाहिए ना, क्योंकि आप लोगों की वजह से हमारे 8-10 साल तो रेगुलेटरी अप्रूवल्स में चले जाते हैं. So in the US, the term of the patent, if the inventor applies to the patent office, that then the term of the patent can be extended. How it is calculated, patent term extension कैसे calculate करते हैं? It is half of the clinical testing time. So if the company required say 5 years of clinical studies, so uska half, यानी 2.5 years, and the FDA required say 2 years. Okay, so 2.5 years plus 2 years, 4.5 years, minus any period during which the applicant was not due diligent. So अगर inventor company ने जो मांगा है वो डेटा नहीं दिया, तो ड्यू डिलिजेंस नहीं किया, suppose 3 months were lost due to the delay caused by the inventor. So 2.5 years plus 2 years, 4.5 years, minus that is 4 years and 6 months, minus 3 months. So 4 years and 3 months के लिए आपका patent term extend हो जाता है beyond the expiry date of the patent. But total extension in the term cannot exceed 5 years. So ये अगर पूरा sum 6 years हो रहा है तो आपको maximum 5 years ही extension मिल सकता है. This is called patent term extension. This is a TRIPS plus provision. So if the patent gets extended because of this provision in the US, then the generic entry gets delayed and the drug does not become cheap for the common public. You can see here, the commissioner of, this is an example of patent term extension certificate. It is says that for this particular patent, we are extending the term by 1,571 days. All this information is freely available on the United States Patent and Trademark Office website. So this is another patent linkages is a TRIPS plus provision. Patent term extensions is again a TRIPS plus provision. Then another provision is data exclusivity. So what it says is, suppose the generic company, it wants to start developing its drug as soon as the innovator company comes into the market. Now, in order to develop its drug, it has to use the clinical trial data because it has to do the bioequivalence. So that data is available with it is freely available on the country's regulatory authorities. So data exclusivity is a monopoly, is a protection which is beyond patents. And it says that if the pharma company claims that it has a 5 years data exclusivity, then for the first 5 years of its provision, the generic pharma company cannot even use its data. Generic company does not want to market its drug. It will be able to market its drug only after the patent gets expired. But data exclusivity means the generic company cannot even use this data, the clinical trial data, for 5 years or 7 years. So that is a data exclusivity. It is a protection of clinical trial data required to be submitted to a regulatory agency to prove the safety and efficacy of the new drug. So this data exclusivity prevents the generic drug manufacturer from relying on this data which is produced by the innovator. It is the period during which the data of the original marketing authorization holder relating to preclinical testing and clinical testing is protected. It provides a form of market exclusivity outside that provided by patent rights. This is not a part of, this is a kind of trade secret. So it does not be, it remains a trade secret for 5 years or 7 years. So these are the 3 TRIPS plus provisions, and the countries are not, it is not mandatory for the countries to give TRIPS plus provisions. So those countries which are developed and they want to protect their industries more than their people, they give this TRIPS plus provision. So obviously you will find these provisions in the US, Europe, Japan, Australia, Singapore, such countries. There are some other abusive strategies that the pharma companies use. One of those is patent evergreening. So as soon as the patent comes to an expiry, as the expiry date approaches, the parent company does a trivial change in its drug and they'll file another patent. So for example, venlafaxine is an antidepressant agent. So suppose venlafaxine का patent आपका खत्म हो रहा है, expire हो रहा है in 2020. So the innovator company in 2018, 2 साल पहले, actually there is a metabolite of venlafaxine, desmethoxyvenlafaxine. And this is a metabolite which actually is useful. Okay, so we are now filing a patent on this new metabolite in 2018, and we are now marketing this drug and not venlafaxine. So they will file another patent in 2018, which will expire 20 years later. So 2020 में venlafaxine का patent expire हो जाएगा, तो venlafaxine के generics तो market में आ पाएंगे. Lekin this company will market aggressively the new drug to the doctors, and doctors will not write venlafaxine. They will write this desmethoxyvenlafaxine, which is going to expire after 20 more years. So generic drug ka availability utna delay ho gaya. This is called as patent evergreening. This is an abusive strategy, and it is so rapidly used by all the multinational pharma companies that you can check what is the extent. See, in France, 68% of out of 3096 new new molecules which were new products approved, out of 3000, 68% were evergreen. Barely 5% of the newly patented drugs in Canada are breakthrough. Remaining 95% are evergreen. Out of over 1,000 new drugs approved by US FDA between 1989 to 2000, over 3/4, so 3/4 of the drugs were evergreen and only 1/4 were new. But the all the evergreen drugs, as though the patents had expired, their generic versions were available. Because of use of this evergreening strategy, the drugs became expensive. So these are the strategies used by multinational pharma companies belonging to the developed countries to extend the lives of their drug patents and to reap more and more benefits. It is a very profiteering business. New drug development is a highly profit-making business, and they further want to increase their profits by patent evergreening or by giving data exclusivities or patent term extension or patent linkages. There is one more provision in the US Patent Act, which is called Bayh-Dole Act. So what is this act? See, most of the drug development in the US is done by universities. Okay. Now, from where do these universities get funds to develop new drugs? These are all government grants, the NIH grants. National Institute of Health funds all these researches. So it is a public funded research. And whenever they develop new drugs and they file patents on these drugs, so when they make it, when they are getting patents and patents are coming out of research, which is funded by a government agency. So who should own the patent? The NIH should own the patent, or the government should own the patent. But in the US, they have enacted this new act, which is called Bayh-Dole Act. So because of Bayh-Dole Act, the universities are able to own the patent, though the research has come from the government funded money. So it allows the institutions or grant recipients such as universities to hold the title to patents on inventions. So ये universities क्या करेंगे? उनके पास खुद के पास patent है. Though the money spent on the research was government's money, government does not own the patent. The universities will own the patents and they will sell it away to the pharma companies. 60% of the pharma research in the US today comes from the universities. So the pharma companies unse license karenge or universities ko paise denge. So the universities become very rich, the professors become rich, and the pharma companies will do further studies on this drug. They develop and market the drug and they will sell this drug at an exorbitant price, okay, to public. Now it is a taxpayer's money. If it is a government funded research, it is taxpayers' money. So the research coming out of taxpayers, the research coming out of public funding becomes so expensive to the public. But these are all the strategies used by the big pharma, especially in the US, to allow the big pharma companies to profiteer, and they do not consider the well-being or the welfare of the society at all, because drugs, the prices skyrocket for the people in these countries. So I told you what is TRIPS and what are the minimum standards of protection. But there are certain flexibilities in the TRIPS, and it is very important to understand these TRIPS flexibilities, particularly when we are talking about access to medicine in today's scenario, which is a pandemic situation. So though the TRIPS say that there has to be a patent on every field of technology, the life of a patent has to be 20 years. But there are certain loopholes, there are certain flexibilities in the TRIPS, which are the policy spaces for the countries to mitigate the impact of patents. So, patent की वजह से जो drugs expensive होते हैं, especially for developing countries or for poor third world countries, जहां लोगों के पास पैसे नहीं होते मेडिसिन खरीदने के लिए, और पेटेंट की वजह से या ट्रिप्स की प्रोविजंस की वजह से जहां पेटेंट एक्सपेंसिव हो जाते हैं, वहां कुछ स्पेस इन कंट्रीज को मिलता है ट्रिप्स फ्लेक्सिबिलिटीज की वजह से। ये फ्लेक्सिबिलिटीज क्या है? So it is Article 7 in the TRIPS agreement. It says that the protection and enforcement of intellectual property rights should contribute to the promotion and technological innovation and to the transfer of dissemination technology. Forget this. Article 8, which is very, very important, it says that it recognizes the rights of members. Members means members countries who are signatory to TRIPS. So Article 8 recognizes the rights of members to adopt measures for public health and other public interest reasons. So right now, what is the situation? It is a healthcare emergency. COVID is a healthcare emergency. And Article 8 says that member countries can adopt measures for public health and other public interest reasons and to prevent the abuse of intellectual property rights. So agar pharma companies are abusive and they are selling the drugs at an exorbitant price and there is a public health concern or public interest reason, then the member countries can adopt measures so that they can ignore the patents and still make the drugs available. Okay. So, and there is one more, we'll talk about this later, that there was one Doha declaration on TRIPS and public health, where all the developing countries came together in Doha ministerial conference, and they reiterated that each provision of the TRIPS agreement shall be read in light of the object and purpose of Doha declaration. And it says that if the public health is in danger, then patent rights can be ignored. So this is a very, very important flexibility in TRIPS agreement, because of which the access to medicine can be improved in case of healthcare emergencies like we have right now. So these are the TRIPS flexibilities. There is Article 6 of the TRIPS allows parallel imports. Now, what is a parallel import? Suppose the pharma companies sell their drugs at different prices in different countries. So suppose the company sells one drug at say 8 rupees per pill in Bangladesh. Okay. And the cost of the drug is 20 rupees per pill in India. Okay. So then can India buy the drug in Bangladesh and import it to India? Because even after import and even after paying the custom duties and all, we can sell it at 15 rupees here. So company, एक pharma company same drugs बांग्लादेश में बेच रही है 8 रुपीस को और इंडिया में बेच रही है 20 रुपीस को. So can India parallelly import it? Can they buy it in Bangladesh and bring it to India? And if they do it, is it illegal or is it legal? TRIPS agreement says this is allowed. This is legal. Okay. So this can be used to bring down the cost of the medicines and make the drugs available at a lower price. So this is one flexibility, very important flexibility. Then another flexibility in Article 27 of the TRIPS says World Trade Organization members or WTO member countries can develop their own criteria for novelty, inventive step and industrial application. हर कंट्री का नोवेल्टी, इन्वेंटिव स्टेप और इंडस्ट्रियल एप्लीकेशन का क्राइटेरिया अलग हो सकता है. TRIPS does not tell you what is the definition of novelty, inventive step and industrial application. Okay, so if it does not tell you what is novel, then the country's patent act can specify what is not novel, what is not patentable. I told you the entire list of inventions which are not considered patentable in India, that is allowed. It is a flexibility. Then there are certain general exceptions. One is called Bolar exception. So what is a Bolar provision? This is called research exemption. So a pharma company, if there is no data exclusivity, then ideally the generic company cannot start drug development unless and until the patent of the innovator expires. But we don't want that. उससे क्या होगा कि पेटेंट एक्सपायरी होने के बाद जेनेरिक ड्रग डेवलपमेंट शुरू होगा और उसके बाद 2-3 साल के बाद जेनेरिक मार्केट में आएगा. We don't want that. We want the cheaply priced generic drugs to be available in the market market as soon as the patent expires. So TRIPS agreement says कि research के लिए ये लोग during the life of the patent innovator का drug use कर सकते हैं. So generic company can use innovator's drug research, they can't sell it. So the generic product they can't sell before the patent expires, but they can develop the generic product before the patent expires. This is called BOLAR provision. Then the most important provision, which is right now has become very important, is the provision of compulsory licensing. Article 31 in TRIPS says that in case of healthcare emergency, a country's government can grant a compulsory license. What is a compulsory license? So, अगर Gilead का patent है remdesivir पे इंडिया में, तो it is a non-voluntary license. There is an emergency, there is a healthcare emergency. So some Indian generic company can apply for a compulsory license to the patent office, and if the license is granted, they don't have to ask for permission by Gilead, and though the patent is on, the Indian generic company can start making the drug ignoring the patent. Okay, this is called compulsory license provision, and which has become very, very important in the today's pandemic situation. This provision is called compulsory license for export purpose. So what is the compulsory license for export purpose? So the compulsory license is रेमडेसिविर पे इंडिया में कंपल्सरी लाइसेंस ग्रांट हुआ है किसी इंडियन जेनरिक कंपनी को, तो the generic company can make the drug ignoring the patent, but it can sell it only in India. It cannot export. But if the compulsory license for export purpose is granted, तो ये किस लिए है? Suppose there is a African country जहां पे कोई हेल्थ केयर इमरजेंसी हुई है और वहां का जो ड्रग है वो पेटेंटेड है, तो दे वांट टू इग्नोर द पेटेंट्स एंड मेक द ड्रग, बट उस कंट्री में कोई फार्मा इंडस्ट्री है ही नहीं, so there is no capability of making the drug. But so even if they grant a compulsory license, who will make it in that country? So in that situation, a compulsory license for export purpose is granted. So अगर इंडिया से उनको वो ड्रग मंगाना है, so India grants a compulsory license on that drug for export purpose, as well as concurrently that African country will also grant a compulsory license on the same drug, and then it can import that drug from India. So there are two types of compulsory licenses. One is for sell in that country and the other is for export. Another provision is government use provision. So government can just notify in the Gazette that here onwards the patent rights are ignored because we want to use this product for government use. So this is another provision which can be used by a country in case of healthcare emergencies. Okay, so during 1990s there was a very bad pandemic, AIDS pandemic, and the African sub-Saharan Africa was hit by these countries. Now in 1995, the antiretrovirals became available, but they were exorbitantly priced. The cost of developing, a cost of 1 year therapy was around 15,000 USD, and millions of Africans were dying. The drugs were patented. The average income of African is 3,000 to 4,000 USD per year, and the cost of these drugs were 15,000 USD per year. Because the African countries did not have any patent laws and compulsory license, now these provisions of 31 bis, that is compulsory license for export and all, they came after this African AIDS crisis. Till then, Africa did not. Okay, and when they tried to, the African governments were pleading to the American and European companies who had developed the antiretrovirals कि please हमारे रोग मर रहे, आप हमारे लिए humanitarian grounds पे आपके drug, and millions of people died. You can see here, this is a graveyard during the AIDS pandemic in Africa. Similar situation we saw the pictures of cremations in India being printed in Wall Street Journal and New York Times. What you see here is the only flourishing business during those days in Africa was of making and selling coffins. Several small kids were orphaned because both their parents died. And this was a big problem of who will take care of these orphan children in the Africa. So it was a very bad situation, and still the drugs were not made available. There was no access to medicine. When these countries tried to amend their laws to be able to export the drugs from India, because before 1995, the Indian company Cipla had developed the antiretroviral cocktail, and they were ready to supply it for 350 USD per year. But they were not allowed because the laws were not in place. And there was a tremendous pressure on the African countries by the big pharma and the US. 39 American and European pharma companies filed suits on the government of South Africa and Nelson Mandela to stop them from changing their laws and incorporating the compulsory license provision and the parallel import provision. So after this, suddenly the WTO and WHO realized that overuse of patents is going to hamper the access to medicine, and especially if something like this happens, some emergency situation happens, then it would not be possible. It would not be possible to make the drugs available, and so certain changes were made in laws. After this, the Doha Declaration happened. So though the provision of compulsory license was there, the small countries who do not have much economic muscle power, when they tried to amend their laws, they were tremendously pressurized. So in 2004, all the developing countries gathered together in the WTO ministerial conference at Doha, and that they declared that we reiterate that we have the right. The paragraph 4 of the Doha Declaration mentions that we agree that the TRIPS agreement does not and should not prevent members from taking measures to protect public health. Accordingly, while reiterating our commitment to the TRIPS agreement, we affirm that the agreement can and should be interpreted and implemented in a manner supportive to member rights to protect public health. So it mentions 4 patents, especially to promote access to medicines. Now, so this AIDS pandemic in 1990s in the sub-Saharan Africa made several changes. Several countries made several changes. After this, several compulsory licenses have been granted, and it was the Indian generic companies who saved the lives of millions of Africans. I am happy to tell you that I have recently authored a novel on, it is not a fiction, it is a true story, and I have written a novel on role of Indian company Cipla and how they saved lives of millions of Africans and what politics going on behind the scenes as far as access to medicine was concerned. The book is in Marathi by the name of Ashini Exunza and has been recently published by Rajhouse Prakashan. So also Doha Declaration says कि वो country the situation actually circumstances of extreme emergency, the country will decide. So it can be a HIV/AIDS pandemic, or it can be tuberculosis or malaria or other epidemics. That the country will decide that what constitutes a healthcare emergency. So compulsory licenses, you might have realized that today in COVID, countries are discussing a lot about compulsory licenses. But since 2001, basically after the AIDS crisis, compulsory licensing of medicines has been used 34 times in 24 countries. Okay, so basically they were on ARVs, that is antiretrovirals, but some compulsory licenses have also been granted on drugs which are not ARVs. The provisions in Indian Patent Act. if there is a healthcare emergency or right now in COVID, what provisions India can use to improve access to medicine. So India, of course, has a very powerful section on compulsory licenses in the Indian Patent Act, which is called Section 84. So Section 84 says कि India में compulsory license कब grant हो सकता है? After 3 years from the date of patent. Okay, so if remdesivir का patent पे अगर आपको 2021 में compulsory license grant करना है, तो patent must have been filed 3 years before. Now, unfortunately, remdesivir patent is filed in India. The last patent filed in India is in 2019. So, in case of normal compulsory license, an Indian generic company has to apply for a compulsory license, and if the government grants it, then that company can start making the drug, ignoring the patent. But they can apply it only after 3 years. What are the grounds on the basis of which a compulsory license can be granted? Number one, failure to meet reasonable public requirements. So now what are reasonable public requirements? That the drug is not available. The vaccine is not available. So it is the public requirements are not met because the company does not have the infrastructure to manufacture the drug in that quantity. So we are granting a compulsory license. Or kab kar sakte hain grant? That the inaccessibility of the patented invention is not available at a reasonably affordable price. The price of the invention is very high. Remember, compulsory license can be granted only on pharmaceutical products, not on any other. Okay, so these are the special provisions related to medicines. So, अगर public का requirement satisfy नहीं हो रहा है, या फिर वो drug बहुत expensive है, or the patent is not worked in India. Working यानी क्या? The drug is not being made in India. Patent India में file किया है, लेकिन drug बना रहे हैं US में और वहां से import कर रहे हैं, वो नहीं चलेगा. You have to make the drug. When the company starts making the drug, making the patented invention in India, it is said that the patent is worked. So non-working of a patent in India is also a ground for grant of compulsory license. There is a jurisprudence in India of grant of compulsory license, and India granted its first compulsory license in 2012 on a drug called Nexavar. So this is Bayer's. Bayer is a giant German company, and Nexavar is sorafenib toside. It is an anti-cancer drug, very useful on kidney and liver cancer. So this drug was exorbitantly priced. The cost of this one treatment cycle was 3,20,000 rupees. And the drug was also not available in enough quantity. The patent was not worked. The drug was being imported from Germany. So on all these three grounds, India granted its first compulsory license on Bayer's Nexavar to Natco Pharmaceuticals, which is an Indian generic company based in Hyderabad. They started selling the drug at 10,000 rupees. So see how much the access was improved. 3 lakh ka drugs milne laga 10 ya 12,000 rupaye mein. Okay. So this, and in this case, the price, when the compulsory license is granted, the price is fully controlled by the government. Now, in case of remdesivir, a compulsory license was not granted, and therefore the companies were allowed to set the prices on their own. There was no role of government. There is a technical issue that though compulsory licenses are legal in Indian Patent Act, though they are allowed by TRIPS agreement, but as soon as you grant a compulsory license, there would be tremendous pressure on the Indian government. There would be trade sanctions as well. Secondly, a compulsory license cannot be granted according to the court if the patent is not older than 3 years. So within the first 3 years, compulsory license cannot be granted. And many applications of COVID drugs have failed because they were filed within the last 3 years. Now another provision is section 92. हमने यहाँ पे देखा कि सेक्शन 84 के लिए इंडियन जेनेरिक कंपनी को कंपल्सरी लाइसेंस के लिए अप्लाई करना पड़ेगा और वो सिर्फ 3 साल के बाद कर पाएंगी. बट अगर 3 साल पहले you want to invoke a compulsory license, like in case of COVID, So the government directly grants a compulsory license and then directs some pharma company. So in first case, the Indian pharma company will apply for a CL. In the second case, according to 92, the government will grant a CL without any application because there is a national emergency or there is some circumstance of extreme urgency or the government wants it for a public non-commercial use. Now, difference kya hai? Yahan the company will ask for a CL. Here the government will grant it. Here the company cannot ask for it in first 3 years. Here there is no such time cap and the government can directly grant. Then 92A, as I said, is a compulsory license for export to other country. We talked about it already. Then some other provisions are according to section 66. The government can straight away revoke the patent in public interest. Section 100, the government use provision. The central government just has to publish a declaration and in the Gazette and conquer the patent for government use for 100
और उसके बाद वहां जाके साइन किया लोगों ने, सबने बीसी ऑफिस जाके साइन किया.
So according to section 102, the central government can acquire. This is 100. This is 100. So Indian Patent Act has so many provisions. What are the provisions? Section 84, compulsory license to a Indian generic company. Section 92, compulsory license by government. Then compulsory license for export. Revocation of public patent in public interest, government use provision, and acquisition of patent by the central government. All these provisions are legal provisions and they could have been used, but India did not use any of these provisions to improve access. Why did they not use? Because if you use these provisions, there are several diplomatic issues and there is an immense international pressure. There is a more harm than benefit to India's global image. आज तक इंडिया ने सिर्फ एक compulsory license grant किया है, वो Nexa पर. And when we granted this compulsory license, India has faced a huge backlash and international pressure. So rather than that, what we have done, the pharma companies like AstraZeneca, they have given a voluntary license to Serum Institute of India. So we did not have to compel. It was not a compulsory license. The company gave the license on its own. Okay. Or for Remdesivir, Gilead gave license to several Indian pharma companies, like 6 pharma companies, Cipla, Jubilant Life Sciences, Hetero and Mylan. In सबको voluntary license दिया. Generally, if grants a voluntary license, then you have to give a royalty for some 4-5% of royalty. But Gilead has given a royalty-free voluntary license. This voluntary license is not only for sale in India, but it is also to manufacture and export to 127 countries. So, अगर प्यार से voluntary license हो रहा है, तो why to go for these harsh provisions which were earlier done? So, in case of Covishield or Sputnik का voluntary license, Dr. Reddy's को मिला है. So, all these were voluntarily licensed product. Compulsory, Supreme Court actually asked the Indian government that why don't you grant compulsory license? But the Indian government said there is no need to grant a compulsory license because the license have already been granted. But at the same time, there is a very contradictory behavior by the Indian government that when asked that why it is not granting a compulsory license or why it is not using all other provisions in the patent law, it said there is no need. But at the same side, you must have heard about this WTO TRIPS waiver. Okay, so India and South Africa have jointly proposed that on all COVID related technologies, all forms of IP, it's not only patents, trademarks, copyright, trade secrets, all should be waived for the period of 3 years. Okay, and this is a proposal for which India and South Africa are pushing. So ये क्या हुआ कि हम इंडिया के लिए कोई भी प्रोविजन यूज नहीं करेंगे जो ट्रिप्स में अलाउड है. ट्रिप सेज दैट यू करो आप ये, तो लेकिन वो हम नहीं कर रहे. बट वी आर सेइंग कि इन जनरल हर कंट्री में हर चीज कोविड रिलेटेड हर टेक्नोलॉजीज के लिए फॉर नेक्स्ट 3 years the trips should be waived off. Okay, now governments are taking action to suspend patents during the COVID-19 pandemic and to ensure that everyone everywhere has access to life-saving medical tools. Now, this proposal by India, so the last meeting happened on this 8th of June, this month, just last week. So this TRIPS waiver has been supported by 100 out of 164 members. But obviously the members who are opposing are the members who are the manufacturers. Like Australia has opposed, Japan has opposed, Europe is very strong. Angela Merkel, you might have heard her opposing the TRIPS waiver very loudly. Okay, but fortunately, to our greatest surprise, the biggest opposer of such kind of TRIPS waivers has been US. But this time, US has supported India's proposal to waive off TRIPS agreement. So India has backed. But what India and South Africa for TRIPS waiver for all COVID-related technologies, but the US has said that we will support only for vaccines, not for small molecule drugs, not for ventilators or testing kits. Uspe nahi karenge. We back the proposal only for vaccines. So it is a very, very clever diplomatic move by America. Why? Because even if they give away the patents on vaccines, what you need, because vaccines are biological products, and in case of biological products, the patents, these are large molecules, so the patents are not enough. What you need is the know-how, what cell lines to be used, how separation and purification is to be done, and all this information is not there in the patents. And without knowing this know-how, without sharing this technology, it is impossible to replicate or reverse engineer the vaccines. It is very easily possible in case of small molecule drugs, generics can be done, or the technologies can be replicated. But, and what we have asked for is the TRIPS waiver on all the technologies, including devices, small molecules, and vaccines. But US has backed only vaccines because they know that even if they stop these patents, it would be of no use. We would not be able to reverse engineer it. And the second use, as I said, is not of patents. India had so many provisions in the Patent Act which they could have used. The problem is of manufacturing capability and availability of raw materials, which is not there. So what will you do with the patents? What will you do with the vaccine patents? Because even if the vaccine patents are freed by Pfizer and Moderna, you would not be able to make it because you don't have the manufacturing capability. Neither do you have the availability of raw materials, because they would be they are blocked by companies. So this is how it is happening. So so many countries have backed, EU has still not backed. So though US has backed, Australia, Japan, and Europe are strongly opposing. China and Russia have supported. So on the June 8th meeting, it is decided that text-based negotiations will now start on 17th June, which means every sentence in India and South Africa's proposal would be negotiated. So what I feel as a researcher in this area, what I feel is even if the patents are dropped on vaccines, it would not be of much use unless the patents on small molecules and technologies, that is test kits, are dropped. But still I feel it is a symbolic win, and it is an answer given by the developing countries to the big pharma that for our countries, the lives of our people are more important than your patents. And it is a move by the developing countries to support access to medicine in this COVID-19 pandemic. So I feel I should stop here. I must have already exceeded my time. So that's all about how the patents are becoming a barrier. What are the provisions available in to the other countries, and particularly in India, and how developed countries exploit the provisions of patents by either exercising TRIPS plus protections or by way of patent evergreening or by enacting laws like Bayh-Dole Act. So thank you so much, and I'll stop here. Somebody asked for my mail ID. I am typing it over here.
Thank you, madam, for such a wonderful session. I can see many comments. I'll read some of them. Dr. Amol Joshi, very nice and informative session. Then somebody, Dr. Rajeshwar has congratulated for your book. Okay.
Yeah.
So lot of full of information about patenting, informative session. So indeed, madam, that was a very wonderful session. You updated us with the latest information, especially for COVID-related drugs also, the patenting related to it.
Yeah, because this is a very happening and evolving jurisprudence, and the latest information was on June 8th, which is last week.
Okay, but still, yes. Madam is extremely thorough in her subject, and she is continuously keeping her updated. So I would now request Dr. Maria Madam from YB Chavan College of Pharmacy to conclude the session. Dr. Maria Madam.
Ma'am, you're muted.
Madam, unmute yourself. Maria Madam. Please unmute yourself. She is telling she is unable to mute herself.
You must have.
Yeah, thank you.
Yeah.
Hello, am I audible?
Yes, yes, madam.
Good afternoon, everyone. First of all, I would like to thank all the team members of Government College of Pharmacy, Aurangabad, for organizing such a wonderful STTP on advances in pharmaceutical research. Today, we had an honor of listening to an eminent speaker, Dr. Mrudula Bele. Madam has explained the intricacies of patents, which otherwise is a very difficult subject to understand. She explained the basics of intellectual properties. She nicely explained the patent, what is patents, and what are the various myths related to the patent. And she broke various myths which are related to the patents. Her talk covered a very important topic, what is patentable and what is not. And she explained the patentability of life forms by quoting the examples of Chakravarti microorganism, transgenic mouse, and Merca gene. She also briefed on patentable natural products and what is patentable in India and what is not. In her session 2, there is access to medicine and COVID-19. Are patents a barrier? Madam started her session with briefing on the process of drug development and explained patent linkages in the USA. Madam talked about the Paris Convention 1995, and she also talked about Indian patent law before 1970 and Indian patent law 1970 and how this law has boomed the pharma industry. She briefed about TRIPS agreement in 1995 and explained the various provisions of TRIPS agreement. She talked in detail about patent linkages and how generic market is linked to the expiration of the patent. She demonstrated how to use the Orange Book and how Orange Book can be used to look out for the patent linkages. She explained the term extension and how generic entries get delayed. She talked about data exclusivity, which prevent generic player to rely on the data patented. She spoke about abusive strategies like patent evergreening. She very nicely explained the various TRIPS flexibility, especially Article 8 and Doha Agreement, and how this can be made. This can be used to make the medicine available during the healthcare emergency. Her talk was supported by various examples and case studies. The session will definitely help Madam all the delegates in their academic research. Your talk was wonderful. No words to, you are a very good orator. It was really a great honor to listen to you. Thank you so much. Thank you.
Thank you
Mayura
so
madam?
much for your kind words, madam. Thank you.
Thank you so much.
Thank you, Maria madam. I would now request Navle sir to give the e-bouquet as well as display the certificate as a token of love and appreciation from our college to Bere madam. Thank you,
so
madam.
much. I accept this virtually. Thank you.
Yes. Thank you so much. I also request Navle sir to display the certificate to Maria madam for the chairpersonship. That's for Maria madam. Thank you.
Thank you. Thank you,
Thanks
sir.
a lot.
Thank you, madam. Thank you.
So, dear participants, I declare that the day one of this first day of STTP is concluded, and tomorrow we will meet at 10 o'clock sharp. You may join 10 minutes before. So, sharp 10 o'clock, we are going to start the first session. So, stay